delegates. We welcome you all to the afternoon session of this conference. Now, it's our privilege to welcome Dr. Ankita Pandey ma'am and Mr. Vipin Saini sir to the stage to be the chairpersons of the session.
Dr. Ankita Pandey is the Science Policy Advisor for People for Ethical Treatment of Animals, India. And Mr. Vipin Saini is the CEO of Biological Agri-Solutions Association of India. I now request the chairpersons to introduce the speakers for this session.
May I request all the speakers, Dr. Jan, Dr. Pradeep, Dr. Deshpande. Pradeep, can you come over? We'll save us a lot of time. A very good afternoon, ladies and gentlemen, both present in the auditorium and online. It's a lovely afternoon, and I hope that everybody is still more energetic after the beautiful luncheon that was just organized by the organizers. It's a privilege to be here standing before you to address the International Conference on Alternatives to Animal Experiments, which incidentally also commemorates with the 6th annual meeting of the Society for Alternatives to Animal Experiments in India, hosted by the Department of Pharmacology, Aligarh Muslim University, Aligarh, and it's a privilege to be here, and thank you, Aligarh, for having us over here. Before I introduce the speakers, I'll just like to say a few words from a few observations that I'd like to share. The chair, Dr. Y.K. Gupta, had earlier stated that we were a MAD signatory, EU MAD signatory way back in 2011, and in 2022, we have been renewed that membership for another 10 years. Thank you very much for bringing that to our notice. And I've been interacting with Dr. Y.K. Gupta since he was the head of the Department of Pharmacology, AIIMS, New Delhi. Another quote that caught my attention was from our UK guest. If I'm not mistaken, her name is Savita Newton. Yeah. So thank you very much for the lovely quote that you shared on Ralph Waldo Emerson. There are always two parties, the party of the past, party of the future, the establishment, and the movement. And that's exactly where we stand as regulatory authorities in the country, where we stand. There is there are the pros and the cons, and there are the yeas and the nas, the yeas. So, I mean, some of the regulators are of an older thinking. Some of the regulators are more energetic, but I have the privilege of stating that we have two such regulators, one, a former Indian regulator, Dr. T. Congo, amongst us, who was a medical toxicologist at the Secretariat of the Central Suicide Board Registration Committee. And we also have Dr. S.K. Jain, who is presently a consultant pharmacologist, a medical toxicologist to the registration committee, who have both have got a vision of adapting new technologies and also seem to encourage their adaptation in this regard. Savita had also mentioned about the future is artificial intelligence. I remember with my short stint on toxicology at the Shri Ram Institute for Industrial Research, when I left in '97, they had just attained a software which was a computerized predictions or mathematical modeling of toxicology of similar molecular structures of various chemicals which you could actually estimate in this regard. So the other thing which I would like to really appreciate is PETA for starting the Dr. Dipti M. Kapoor Endowment Award. I had an opportunity of working with her on the toxicological protocols for the registration committee, which was incidentally also drafted preliminary by Dr. D. Congo. And it's a wonderful move by PETA and really a lovely gesture. I really do appreciate that thing. Ladies and gentlemen, my name is Vipin Saini. I happen to be the CEO of Biological Agri-Solutions Association of India. I had a brief stint as a toxicologist before I moved on into the field of regulatory affairs, and I'm considered a specialist in that regard. Our organization are manufacturers of both biological agri inputs in particular, including biofertilizers, biopesticides, and biostimulants. And recently, biostimulants regulations have been regulated under the Fertilizer Control Order with both acute and ecotoxicology. And that's one way also where we look at the alternatives in this area. So not taking much of your time, I'd like to introduce my co-chair, and or what may I say, chair. We've divided the chair amongst ourselves. So Dr. Ankita Pandey is currently serves as the senior advisor to PETA India, where she works for various regulatory agencies to promote the regulatory acceptance and implementation of non-animal methods. She completed her PhD from CSIR Indian Institute of Toxicological Research, Lucknow, is a recipient of a prestigious Tulsa Bhai Educational Trust Award by the Indian Academy of Neurosciences, and the author of a book, Footprints of Toxicology in India. It's a wonderful book. Those who have not read it, I'll strongly suggest that you guys come across. She will be giving a talk on the opportunities and challenges for implementing 21st century non-animal toxicity testing in India. Ladies and gentlemen, Dr. Ankita Pandey.
Oh, thank you, Mr. Saini, for such a kind introduction, and good afternoon, everyone. At the very outset, I would like to thank the organizers, especially Dr. Rahman, for giving PETA India an opportunity today to organize such an important session. Before we dive into the session, I would like to introduce everybody to PETA India. We are basically an animal rights organization. Our primary area of interest is animal protection, but it's also to promote good science. And PETA India scientists have been working very closely with government regulatory bodies, with companies and scientists to promote non-animal methods of research and testing. We're also members of the PETA Science Consortium International, which is an international group of around 25 scientists from 9 different PETA entities. And we work to advance reliable and robust non-animal testing approaches all around the world. As you can see, we have stakeholder status with several scientific organizations, and we work towards global harmonization. Some of the ways how we do this is by funding the development and validation of new test methods. We organize training and workshops and webinars to create awareness on the use of non-animal approaches. We publish and present at scientific conferences, organize expert working groups to strategize ways on how we can reduce animal testing, and also conduct retrospective analysis along with regulatory agencies that help shape guidance documents. So getting back to the session, the focus of today's session is on toxicity testing of agrochemicals and industrial chemicals. And as we all would know that animal testing has been like the gold standard for regulatory decisions for many years now. And basically, there is a battery of tests that is required, ranging from acute studies to long-term studies to assess whether these products could basically harm humans, wildlife, or the environment. But this current approach is currently faced with a lot of challenges. For example, animal tests, they're known to have certain limitations. For example, they are not very reliable, they're not reproducible, they are cost and time intensive, and they also lack human relevance. Moreover, current tests do not even reflect the most up-to-date science. In fact, some of these methods, which are based on animal models, they date back to more than hundreds of years. What's also concerning is that these tests are quite animal intensive. So, for example, it's estimated that more than 10,000 animals they use to test just a single chemical pesticides. And as we speak today, there are thousands and thousands of chemicals which still remain untested. And this current approach based on animal tests, it's highly unlikely to meet increasing demand of chemical assessments without a concomitant increase in the number of resources and animal lives. So this need for a paradigm shift to have methods which are faster, they're reliable, they're fit for purpose, and human relevant have led to the development of new approach methodologies, or popularly being referred to as NAMs. The definition varies as per which regulatory agency you're speaking with, but these are generally defined as any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment and avoid the use of animals. What's important to note here is that NAMs may not necessarily be newly developed methods, but it's their application to a particular regulatory agency decision-making process which may be new, or replacement of a traditional animal test method that may be new. So these are some of the NAMs that you usually find within a NAMs toolbox, which are either being used standalone or they're being integrated together to make health protective decisions. So if we talk about the international scenario, we have regulatory agencies that are recognizing the importance of timely uptake of NAMs to prioritize the use of human biology-based methods and reduce reliance on animal testing. For example, the Environment Protection Agency in the US that regulates pesticides and chemicals, they have a strategic vision in place for adopting NAMs, which is aimed at even replacing many traditional animal-based methods for chemical testing. EPA has a very long-standing history of applying a weight of evidence approach while giving out waivers for several endpoints. And this approach has actually saved a lot of money. As per the publication from 2012-2019, EPA has been able to save as much as 300 million in study costs, 6 million in study review costs, and more than 2 lakh animals. So in addition to promoting good science, animal welfare, there are a lot of economic benefits also that are associated with the use of these NAMs. EPA, in fact, even has a work plan published where they're describing strategies and objectives to reduce the use of animals through the use of NAMs that can offer the same or even better protection than what animal models give to us. So this work plan highlights the importance of identifying data gaps, developing new methods, establishing confidence in them, engaging with stakeholders. In fact, since 2019, EPA has been holding regular conferences to discuss the state of the science with respect to the development of NAMs. Taking a quick look at Europe in 2021, the members of the parliament, they passed a motion for resolution to phase out the use of animals for research, for testing, and for education. And this resolution requires the European Commission to basically accelerate the development of new NAMs, to have targeted funding for them, to create more awareness, and even work on enforcement issues. And just last year, we had the European Chemical Agency commit to developing a roadmap to again prioritize the use of NAMs and end reliance on animal tests. A similar progressive move has also been seen from the European Food Safety Authority, EFSA, and even in the UK, where everybody is committing towards developing new roadmaps on how they can accelerate towards animal-free science. Other regulatory bodies such as Canada, in fact, even we have a lot of publications from global companies that are voicing similar opinions and are advocating for a science-informed decision-making rather than a checkbox approach to conduct risk assessments. So it's very, very clear that, NAMs, in addition to offering faster and more efficient toxicity testing, they have the ability to fundamentally transform today's regulatory testing paradigm. And India is faced with a similar opportunity to reap the benefits of the 21st century science. So the idea is to basically move away from an animal-centric checkbox approach towards a 21st century risk assessment that's fulfilled using a weight of evidence approach that takes into consideration all the existing information. You consider the use and exposure scenarios, you accept data from non-animal testing strategies, and animal testing be conducted only as a last resort. Again, NAMs do have the potential to offer the same or even better protection to humans' health and environment while being human biology based. So we are not talking about compromising human health or the environment in any way. We're talking about methods which are at par or even better than animal models, and they're human relevant, they're faster, they're cheaper, they're more efficient. But we understand that with these opportunities comes a lot of challenges. Certain translational barriers exist, whether it's from a regulatory acceptance point of view or from an industry implementation perspective. And this is where we work to bridge the gap. To address this, we have put forward certain key steps as way forward that can be implemented and can help accelerate this transition towards non-animal methods. I would really love the opportunity to discuss some of these ideas during the panel discussion. So as you can see, we have a very impressive lineup of speakers today, and I'm hoping that with this session that brings in perspectives from different experts, we can actually brainstorm over our current challenges, opportunities, strategies that can help overcome some of these barriers, and we can outline some key steps that can help us accelerate this transition towards animal-free safety assessment. So to conclude, I would just like to say that replacing animal tests does not mean that we are halting scientific progress in any way or compromising protection of humans and the environment. Instead, implementing non-animal modern approaches will improve the quality as well as the humanity of our regulatory science. Thank you so much.
Thank you, Ankita. A very impressive talk, and definitely I know your enthusiasm for promoting NAM. And thanks for giving us a brief on the US and the EU roadmaps towards reducing animal use in experimentation. Ladies and gentlemen, I have been interacting with Dr. Ankita for quite some time, and during our advocacy parts with the ministry at various forums, she's very particular that a particular phrase has to be written, the wording needs to be appropriate. And that's where you'll find new guidance documents coming across with exactly the same phrases, which have far more meaning to it beyond what is actually being articulated. Our next speaker I'd like to introduce, I'll be skipping the queue, but I'll be calling upon Dr. Abhay Deshpande. Dr. Deshpande completed his doctoral research in the field of microbial fermentation at the prestigious University Department of Chemical Technology, Mumbai, then known as the University of Mumbai. Having led JRF, the JAR Research Foundation, over the past 14 years has enabled him to steer JRF into the league of one globally respected major player in the GLP contract research organization for resolving the regulatory demands for registration, and he has now dedicated his efforts to defining the vision of JRF for the next decade. He's been instrumental in driving the strategy for establishing and guiding the dedicated R&D facility, wherein 5% of the global revenues are invested in creating new research avenues in compliance with the trends in science, utilizing NAM tools like silico and in vitro in safety assessment, especially with application of spheroids and potentially application of MPS-based solutions using sphericides. He'll be talking on NAMs. We have some, we need some more. Ladies and gentlemen, Dr. Abhay Deshpande.
Good afternoon. Thanks to AMU for making arrangements for this conference. Thanks to PETA for inviting me for this talk. To be honest, she made my life very easy. She has spoken a lot about most of the things which I was going to. So that actually gives me some time to speak about some other things. So this is the quick agenda. I wouldn't spend time on that, but primarily we are going to be talking in and around NAMs. The only difference, Ankita, is going to be something where I'm going to put a few challenges in front of you, which I'm sure you will take to Nicole, you will take it to the other lady, and we should find a solution for these things. So just to quickly go over my understanding of NAMs, or as a CRO, how we look at NAMs. We realized about 5 years ago that there were frequent requests for in silico interpretation, and that was typically coming for the mutagenic impurities or degradants of a product. And we realized that there's going to be a kind of a field which is important, and we were thankful to have one gentleman, Dr. Bharat Rudresh, who joined us about 2 years ago, and he has established a full-fledged department. Now, typically the kind of things that happen when you do the in silico analysis is multifold, because globally, thanks to NTP, thanks to the European Union and the people there, the database is being enriched every day. Every day, a new chemical with known liabilities or challenges are being uploaded there. Once this is being done, that becomes a source of a database. So whenever Bharat has a challenge to look at a certain chemical, whether this is likely to have a similar challenge or not, he's able to come to that. And that's something which, using QSAR, read across, pharmacophore modeling, MD simulation, so on and so forth, this looks pretty easy. Only 4 different terms I have used. But I know that the guy uses about 12 different tools at a time, and more often than not, because the data does not come out comprehensively clear, he tends to use multiple tools to come to a kind of a reliable data. This can be performed with human systems, but we were surprised when a client request came. We could do it for plants to find out metabolism or how the plants interact with a given fungicide or a herbicide or something. We were able to find out how it could act in a human being. So such work has been giving us this kind of a capability. And as you can see down there, I have listed a few things that we do. I'm not here to talk about JRF. Primarily, the idea is to tell you about the benefits of NAMs and the challenges of NAMs. With this in silico tools, we have established a set of parameters which can be extremely useful for eliminating challenges which are likely to come with new lead molecules or lead optimization process, etc., etc. It can also be used for the old challenging molecules. We see this very frequently with the REACH requirements. REACH is a European requirement for the industrial chemical registration, and that requirement typically calls for, suddenly they come out of the world and they say, okay, we find this particular mutagen and that seems to be toxic to a certain bacterium in soil. Go on and find out whether it really does so. In such case, we go back and check that, and that's a kind of a capability which is what I did. There you go. In vitro solutions. Now, how we look at in vitro, the coordination between in silico and in vitro is, assuming there is a molecule that tells us that this particular product is likely to affect a certain adverse outcome pathway. To make it very simple, I will use an example of an endocrine pathway, also thyrogenic pathways. If it is affecting a certain thyrogenic pathway as per the in silico predictions, we would confirm them with the cells. Now here, typically, what we had been doing at JRF is doing it with the human cells as well as rodent cells, because while Ankita would like to know that the animals are eliminated, there is a huge requirement. That point comes later for you, Dr. Jain, is the regulators still ask for a lot of animals. And that's something where we use this data more or less as a corroboration data. We use typically most of the in silico in vitro methods would use monolayers of primary cells or stably transfected cells or genetically modified cells or lentivirus loaded cells or so on and so forth. And these particular cells would be tested against a given xenobiotic. I'm using a xenobiotic as a term primarily because I don't want to talk about crop care. I don't want to talk about industrial chemical or cosmeceuticals or pharmaceuticals. So it's basically a xenobiotic and how this xenobiotic is likely to affect. And here the approach is very focused. We don't stop at cytotoxicity, no MTT assays, no LDH assays. We go well beyond that to find out, is there a particular pathway? For example, coming back to same thyroid TPO inhibition pathway, is it really affected in the thyrocytes isolated from human beings, or ideally something went wrong? Did I do something? Oh, it's there. So whether that pathway is really affected in a human being as well as rat, and we stop there. And that's something where we are able to conclude how in vitro can be best used, apart from hazard identifications, for which we'll come to it a little later. Did it move? No. Can somebody help me? I think I got some other window here. I don't know whether I made a mistake. So while he is dealing with the technology, the next point I was coming to was about the animal research. Now, typically the animal research as of today has been focused on hazard identification, where, as you rightly said, the acute toxicity tests and so on. And honestly speaking, I don't stop at the mammals. I also take the other life forms, the aquatic life forms, etc., also into consideration, and whether these are affected. Now, these particular, what's the difference between these two is basically in vivo would help us to give a complete organism picture rather than a particular cell picture. So I'll be going about some of the benefits and challenges of NAMs. I'm not going to make you read this, but primarily to make a point very, very simple. The benefits are it can be used fantastically for elimination of hazardous compounds right there at the design stage. So there is no further expenditure for the manufacturing companies to invest into a molecule which is likely to get into challenging situations. The other is typically for checking out the liabilities of a known old molecule or its metabolites in different life forms. There are several available solutions and they are being used, and a lot of commercial as well as free open market kind of situations are available for in silico, and we can use one of them. My only challenge with in silico is 70% is the predictability. So you have 30% possibility of a false positive or a false negative molecule being moved forward. So if left to me alone, would I depend or accept on in silico? I would say I would better get a confirmation on that. And that's where the limitations of in silico are primarily because of this 70% limit. The next is, thank you, sir. Again, you have a long list, but I think I have some time restrictions. So all those nice points that you can see that we can do as benefits, but the limitations which we face, Ankita, are availability of primaries in India. Dr. Akbar Shah had been trying to help us get hepatocytes, and it has not been easy. We tried several sources. That is one part of the story. Many times we get cells which are representative of 2 or 3 or 4 or 5 individuals. More often than not, the typical DMP case studies call for use of 3 different human hepatocytes for certifying a product. Now, do these 3 different individuals represent 7.5 billion of human population. That's a limitation of work with the primaries or the cell lines. Other is when there are stably transfected cell lines like the ones which we have for OECD 455 or 458. These cell lines, and there are large number of parameters which they have defined, have a kind of compliance parameters. We have seen reports of international CROs as well as the publications that there are exceptions taken because some of the parameters do not meet the compliance parameters. And this is a major challenge with these kind of cell lines that we are using. The individual cells. Now, for example, currently we are working on the hepatic spheroids. And what we thought was we will go to make the representation of a full liver. So Priyanka, whom you know, Dr. Akbar, she took hepatocytes, she took Kupfer cells, and she took stellate cells, and they were all individually cultivated. She tried doing co-culture. The representation of all the three was not comparable. So you can, I mean, there are commercial products available where they say we are giving you product which is a liver representing full liver, but it does not, because batch to batch variation of different cells within the purported organ are not the same. Availability of these resources is a problem, and Christian is going to be there. I think he's there online. Christian had been trying to help us since he was in the earlier organization as well as current organization. There is a specific limit within which the cell has to be received on a certain age of a cell in our laboratory, and within X number of days it has to be done. If there is a airline problem, if the consignment arrives on Friday, if there is a customs officer who may not clear the particular consignment, we are lost money. All those 16, 20, 40, 6, whatever we have. Sorry, 2 minutes. Oh, okay. So I need to move. But anyway, these were some of the important points that I was making. Just a few points here. NAM's acceptability. Sponsors are excited. They like it because it saves money. They like it because it eliminates their molecules. They are working a lot and investing a lot in generating this data and submitting to the regulator to get their approvals. Regulators, guarded, cautious optimism. Dr. Chen, apologize, this is not applicable to you, but all the regulators across the globe, except the US and the European regulators, have this challenge that while they say, yes, we want to do non-animal tests, but they surreptitiously ask for animal tests. And this is something which is a serious challenge. CROs like us, we are confused. We want to do it because it is good, it's fast. We don't have to invest a lot of time on conducting the study as it is the animal study. But when a sponsor requests, and if I have to get the cell line from Christian or someone, and if it is going to take 3 weeks, it is the batch is not in manufacturing, I have a chance. So that's one problem that we have. Logistics of shipment is also a challenge because some of them are sent at room temperatures and some of them are sent at lower temperatures. So for us, it's a kind of a time. Ankita, you have this presentation. You're free to share with anyone. There is nothing confidential about it. I'll just make one point here because he gives me the 2 minutes. Quality standards of the NAMs is a serious issue because while you have GIVIM, which is more of a guidance document, we have GCCP1, GCCP2, which is again more of a guidance document where ethically I'm supposed to comply. I can keep my record, but there is no validation verification. And this is a kind of a problem. If at all we want NAMs to proceed, one of the things is globally harmonized, acceptable quality standard has to be established. There are few things about the cell-based assays. I'm not talking about the in silico because I mentioned 70%. Cell-based assays, individual to individual scientists, my lab to the next lab, my lab, next lab to the lab in Mumbai, a lab in Chennai, or a lab in Europe will not have the same result, even if the same cells are shared. So that's a kind of a reproducibility, robustness kind of a problems exist with that. So while it has been, there has been a euphoria, in vitro seems to be a kind of a proposal that is coming through. This was one of the latest papers from Dr. Clippinger, and my questions are here. The burden of establishing human biological relevance is on whom? Authority, who is the independent reviewer? Is it the regulatory or scientific authority? Which authority will assess these cells? Is it a GLP authority? You have the presentation, Ankita, don't worry. He's making me rush. Oops. Can someone, is the screen there? Anyway, this is one point which came from Hartung et al. Again in 2022. I apologize for 1922, it's 2022. Which authorities should, no, he is suggesting, in vitro approach, he's suggesting that we use all this data in a collaborative manner or coordinated manner to determine deterministic or probabilistic possibility of hazard. Now, again, the question is who is going to do this? That's a kind of a question we have. It's a repeat, so I wouldn't spend time on this. I don't need to, but what you must have gathered, what we are doing, we are trying to do in silico, we are trying to do in vitro to confirm in silico output, and if and when required, only within the norms of ALAC and CPCACF, wherever there are permissions available, we do the animal studies. But when we do, what we would like to do is the studies should have, if you are conducting one study and if you're using 100 animals, ensure that you get maximum data out of it. So you cover the endocrine disruption, systemic adversities, DART challenges. There is a EOGRTS guideline for OECD 443. Now that's something which can be extended to get something better. I'm not going to repeat this again. This presentation is available, but these are basically the guidelines which have been released by the regulators. Some of the guidelines. So that's it. I'm sorry, Bipin, I took 1 minute extra or 2 minutes extra. Thank you so much for your patience.
Thank you, Dr. Deshpande. I know you are in a more rush than I am. So anyway, I have a question, and if there's any audience who have got question, we have got time for just 1 or 2 questions from the audience. Just keep yourselves ready. In the meantime, Doc, I'll just like to inform you probably and the audience also that in 2015-16, during the review of the revision of the toxicological protocols under the EU, in India, it was acceptable that in vitro or alternate methodologies will be may be accepted, provided that the industry, and this question comes across to Dr. Pradeep also. Just a reminder that if the industry could provide a data set in vivo along with a data set of in vitro or with listed animals, and therefore for the regulators to compare the I mean, their kind of trust on the alternative methodologies to be evaluated. Doc, I have a question which is structured into 2 parts. According to your talk, that we've been moving forward towards greater integration of novel methodologies, which will also require continuous efforts of capacity building. You've already highlighted that thing over there. Now, as a CRO on an international platform and for guidance in the Indian one, what are your thoughts on how enabled are the present Indian CROs in this respect? 1, and 2, how do we ensure that Indian CROs are well equipped to evolving technologies? I'm leaving out the part of how we can educate the regulators of that part, but these are 2 sets. If you could just.
So I will take the second question first, and while I may be coming here and talking to you, and I do hear here and there that the other CROs are not coming to talk about it, but everybody can do what we can do. I wouldn't say that we might have a few steps ahead of the others because it started way back in 2009. But I think everybody has the capability. So the capacity with CROs exists. It's not that it doesn't exist. Bipin, what was the second one?
The first one was, as a CRO, what are your thoughts on how to enable that Indian CROs to develop this, capacity building?
One of the things which I know, being a CRO for last 20 plus years in this field, initially we were small CRO and we used to do what everybody else does in the world. Now we tend to do a few things which are a little bit ahead of time. The others are not daring to do so because what I mentioned earlier, the guidelines, guidance documents, and quality parameters are not in place. If we have them in place and if they become mandatory, believe me, everybody is going to do it. I don't think it's going to be some. Let's accept it. I mean, we have these students here. Cultivating cells is no big task. I mean, you need possibly 6 months of practice and you can cultivate cells. Running some analysis, again, no big task because most of these are pretty straightforward conventional methods. It's not something that the capacity does not exist. It's only that the people are scared of putting their money. Like, thankfully, you mentioned that we invest 5% of our earnings into this kind of research. Others may be investing, I don't know. But the younger or smaller CROs will not invest their money onto this, which the regulator does not seek. Does that answer you?
Sure.
Okay. Thank you so much. Thank you, everyone.
Thank you.
Thank you,
Hello.
sir.
Are there any questions from the audience that the audience would like to pose in? Come across. Doc, you need to be on stage. Please.
Yeah, go
You need to introduce yourself. Come in front of the mic. Introduce yourself and then raise the question.
Hello, can you hear me?
Hello, this is Dr. Maharajan from Allahabad University. So I have been working on quite some time in zebrafish embryos. So since like alternate to animal models like zebrafish comes under 3R principles and everything. So in India, like recent guidelines on CPCSEA 2021 on fishes. So it mentions basically for fishes about ethical requirement and everything. So in 3R up to 120 hours post fertilization, these zebrafish embryos doesn't require like ethical permission in some European countries and everything. So what is the status in India, whether the embryos needs ethical permission in India? And
I think that question is to you or me? Me?
Means I mean like 3R.
Anyone.
Yeah, anyone.
Okay, one thing I can tell you, gentlemen, the whole idea that 120 hours of life, it is not considered to be in vivo, is basically very questionable. But as of now, we had been doing zebrafish embryo research and we have not been questioned, either by Indian authority or by international authorities. So it's not something which is a big challenge. It's a different piece of consideration whether you're using zebrafish for human-related research or you're doing it for ecological research. But in either case, the age and the in vitro in vivo partition parameters as of today are vague and they will continue to remain vague, and that's good for you.
Thank you.
Thank you. Thank you, Doc. Dr. Deshpande has to leave early and he would like to excuse himself from the conference. May I ask
I now request Dr. Ankita Pandey ma'am to present Dr. Abhay Deshpande sir with a token of appreciation.
Hello, this is Dr. Subramaniam. Can I say something here? Hello? Hello?
Thank you, ma'am. Thank you, sir.
Ladies and gentlemen, our next speaker is Dr. S.K. Jain, the present medical toxicologist in the Secretariat of the Synthesis Insecticide Board and Registration Committee. Dr. Jain, may I have you on stage? Dr. Jain received his PhD in pharmacology from the University of Veterinary Medicine, Hanover, Germany in 1994. From 1983 till 97, he had served as an assistant professor pharmacology and toxicology with the Haryana Agriculture University, Hisar, and later on as a professor at HAU, or what is currently known as the Lala Lajpat Rai University of Veterinary and Animal Sciences, Hisar, till 2018, during which he guided 2 PhD students and 9 MBS students in pharmacology and toxicology. He is a recipient of several awards and fellowships, has 64 research papers to his credit in both national and international journals. With 34 years of post-qualification experience, Dr. Jain is, as I earlier mentioned, currently the Senior Consultant Medical Toxicology with the CIB and RC, Ministry of Agriculture and Farmers Welfare. He'll be talking about applications of new approach methodologies to meet agrochemical testing requirements in India. Ladies and gentlemen, Dr. S.K. Jain.
So first of all, I thank PETA India and the society for inviting me regarding discussion on various guidelines which we have modified recently for regulatory purpose for pesticides. We have brought a lot of changes which help the industry as well to get easy registration of various pesticides in the country, taking into the global challenges and the requirements globally that we have tried to incorporate in our country. So we all know that the CIBRC regulates under 2 Act, I Act 68 and Indian and Insecticide Rule 71 for registration of various pesticides, control the import and manufacturing in the country and sale in the country, as well as it is controlling both advises government of India as well as the various state governments how to go for the further use of the various pesticides in the country. As we all know that India is the fourth largest producer of the pesticides in the world, and we have about, I think, 900 pesticides which have been registered till date today. So far, 62 pesticides have been banned because they are coming in the highly toxic category or extremely toxic category. Pesticides which have been allowed with restricted use is about 9. A lot of discussion is going on for another 20-30 pesticides which should be either banned or should be restricted in the country, because they have been already banned or they have been restricted for use in various countries in Europe, US and other countries. CIBRC has already refused registration for about 18 pesticides because of risk analysis, and that we have found they are not safe and not efficacious. So we did not allow. As we all know that the requirements for registration of pesticides in the country, both human health assessment and we go for the ecotoxicity assessment, that is environmental assessment. So this is the normal requirement for submission of an application for registration of any pesticide. Require, as per the guidelines, various acute experiments in animals, subacute, that may be 1 month to 3 months study, and then long-term studies that are mainly the developmental, reproductive, and chronic toxicity and carcinogenicity. Ecotoxicity assessment is made in birds, fishes, earthworms, honeybees. Now recently we have tried during last few years that let us also accept the new approach methodologies can be allowed or can be considered for registration in addition to the data generated on animals. So in 2017, CIBRC approved a document on toxicology where we have already considered 3R principles, and we allow all these for considering all the methods, including in vitro method, in silico method, and we also consider for waivers for which we have set of guidelines, data breaching concept that has been accepted and we are allowing. These are some of the guidelines for chemical pesticide which were allowed in 2017 for granting registration of various chemical pesticides, and this is given already in the document. Now, recently, few months back, we have already approved the new guidelines where we have given waivers. We have allowed alternatives, but with conditions. Waivers would be considered when existing information provides a robust and full scientifically weight of evidence approach, and which is based on red across bridging, either structurally or biologically related similar pesticides, physicochemical properties, and exposure patterns that depends on compound to compound, case to case basis. Further, we have gone ahead. The replacement alternatives not involving experiments on animals would be considered and accepted where data from valid 3Rs, methods recognized by OECD and other renowned regulatory authorities is provided wherever it is applicable, depending on the case-to-case basis. That we have already allowed. However, there are a lot of conditions and restrictions before we accept the alternatives for registration of the compound. This is available, link is given, and you can have full. This is already we have already loaded. Then we have further gone for accepting the weight of evidence approach, where we can examine all the existing data on the product, on the compound, including epidemiological data or experimental data, which may be animal data, in vitro data, in vivo, in chemical. Non-testing strategy, that is red across or bridging concept from again similar or structurally related compounds, weight of evidence assessment of available data, whether it is sufficient or not, that has to be evaluated and examined thoroughly. Then the data available on studies where animals have not been used, and it has been proposed that animal testing should be the last resort. But again, there is a rider. If the data on replacement alternatives or other things is insufficient, then animal data has to be submitted for consideration of registration. And this is already given. So we have encouraged the registrants that use weight of evidence approach, considering all the existing information on the compound, on the product, and even they can go ahead by discussing in detail with the experts in CIBRC regarding submission of the application. So they can do detailed analysis and then come for registration. Then we all know that grouping of chemicals, that is chemical categories and Red Cross approach, it is given in 2014 document, guidance document using the OECD, that is QSR tools, where we can use this QSAR tools for various chemicals. However, again, it depends on the what are the possible value, whether it gives sufficient results regarding acceptance of the data. So again, link is given, and here we have encouraged registrants to implement all Red Cross or data bridging concept for chemically as well as biologically related compounds. Now in our country, we have tried to encourage the applicants to consider the pesticide use and exposure pattern for a risk-based assessment. So that is exposure driven, oral or dermal or inhalation. These are the common routes of exposure of the compound for toxicity. Now, during last 10-15 years, the OECD guidelines have come for in vitro testing on skin as well as eye, and they can be considered provided we get full data for replacing the animal experiments. That is eye testing in rabbit or skin testing in guinea pig or in rat. That can be considered only when we get the complete information which determines that the product is safe in vitro test for skin as well as eye. Only then that non-animal method will be considered for consideration of registration. All the internationally valid non-animal methods, they can be accepted if they are applicable. Again, we have mentioned skin irritation or corrosion, or eye irritation or eye corrosion tests which have been developed by OECD. Various tests are already available, and we have already given a link. Again, this provides about integrated approach on testing and assessment of the skin and eye. Testing that can be considered and they can be applied for assessment of both eye as well as skin irritation study. Lot has already been discussed by Dr. Deshpande as well as Ankita about QSAR toolbox, and still we are not fully convinced about QSAR toolboxes. There are many which have been not only Derek or Sarah, some one or two more have come to my knowledge, and even I received application providing the data on QSAR toolbox, but I was not fully convinced whether carcinogenicity studies in mice can be accepted only based on some assumptions in QSAR toolbox. At least I was not fully satisfied and I rejected, and so the applicant have again submitted the study he had already might be in archive. So they can help QSAR provided it is the data is fully robust and can be considered. Just 2 minutes, I am winding up. Again, use of integrated approach on testing or assessment, we have already covered. Again, coming to the international guidelines, that already has been covered that various countries, US, North American countries, Australia, Canada, European countries, they have come with these alternatives approach, but still it is debatable whether it can be considered. Still they are considering or not considering in Europe or in US or in Australia or in other countries. It's a questionable whether we can replace testing on animals or not. Still a lot of debate is going on. This is about Canada guidance document, then Australian government, some information is available. Then European Union reference laboratory for alternatives to animal testing, again provide some information. We are trying to promote NAMs in the country, but again, question mark, whether we can directly accept the alternatives or these approaches or not. Still, we are not getting much, not more applications for alternatives or weight of evidence approach, except Weavers, where they give full justification, which we thoroughly examine and consider whether it can be allowed or not for Weavers. So still this is gray area in the early stage. So concluding our talk about NAMs, whether what is our purpose, whether it is reliable, NAMs can be accepted or not for better human health as well as safe environment. Though we know it might be less resource intensive and faster. Presently, there are again lot of debate going on that it involves a lot of money. It is not so cheap as people think. Maybe later on in future might be cheaper. So that if it is acceptable later, then it might be the registration might be faster and our farmers might be benefited quite a lot. This is very important. Third point, we should avoid duplication of tox data generation. Where existing data provides the full information, why to again go for the animal study? That should be allowed, that should be accepted when where the compound is same. We should internationally harmonize the guidelines that we have tried in our recent revision, and we should try to reduce the use of the animals. That will help, as my earlier speaker, they were talking that daily more than 2 lakhs animals are used for conducting the test, that we should think seriously that how we can minimize use of animal and go for the NAMs. Thank you very much. If you have any question, please.
Thank you, Dr. Jan. Are there any questions for Dr. Jan at this point of time? We can take 1 or 2 questions. Anybody? Thank you very much, Dr. Jan. You've been a amazing presenter, and thank you for all your knowledge. Just two things, sir. In the conclusion, if you could also take upon with the registration committee, the implementation of the minor change regulations will help a lot in reducing the toxicological burden and regenerating the whole data on new formulations with minor changes.
I think with the new guidelines that are already available, we have tried to reduce a lot of experiments, a lot of parameters. We have reduced from 2 species to 1 species. We have already brought down number of experiments, and in lot of parameters we are considering the waivers where it is not required to duplicate the studies, or we are giving lot of considerations based
Right.
on weight of evidence approach, and we could consider alternate in future.
Thank you, Doctor.
I
Thank you
think
very much. Please keep us. We'll hold a panel discussion soon after our last speaker, last but not the least, Dr. Pradeep Kumar. Yeah.
Thank you for the session, sir. I would like Mr. Vipin Saini, sir, to give a token of appreciation to Ms. Dr. S.K. Jain, sir. Thank you.
Let me just introduce you for the sake of the audience. Dr. Pradeep Kumar here is earned his degree in veterinary sciences, Doctor of Veterinary Medicine, and Master of Science from Bengaluru Veterinary College. He's after completing his education, he joined Advenus, which is a preclinical CRO in 2006 as a study director for general toxicology and safety pharmacology. Dr. Pradeep is a diplomat of the American Board of Toxicology and is currently working with Corteva Agri-Sciences as a regulatory toxicologist. He'll be speaking upon the topic of implementing norms to modernize agrochemical risk assessment. Ladies and gentlemen, Dr. Pradeep Kumar, floor is all yours.
How do I use this?
First of all, thanks for the organizers, Data India, for providing this opportunity. In this presentation, I would like to discuss some of the general aspects of NAMs from the industry perspective, importance of NAMs in acute toxicity testing and its regulatory landscape, and finally, a couple of examples of potential applications of NAMs in acute eye irritation testing and GHS additive approach. So for well over a decade, efforts have been underway in academic, industry, with regulators, government institutions to develop in silico, in vitro, in chemico methods to assess mammalian toxicity as well as ecotoxicity of agrochemicals. So these methods could be computational modeling, omics applications, high throughput screening, imaging, cell cultures, tissue or organ engineering, advanced imaging or scanning techniques. So all these efforts put together, we can call them as NAMs. So NAMs essentially are any technology, methodology, approach, or combination of that efforts that can provide information on chemical hazard and risk assessment while avoiding the use of animals. So NAMs are replacement of a traditional testing requirements, and they are intended to provide better evidence about adverse effects of different target species. And also they help us to support agencies' regulatory decision-making process. However, please note that these NAMs are not always necessarily a newly developed methods, and NAMs are not designed to become direct substitutes of in vivo methods. So to assess the adverse effects or potential effects of agrochemicals and to establish the scientific confidence, credibility, and applicability in the regulatory decision making, some of the essential elements of NAMs are fit for purpose, human biological relevance, technical characterization, independent review, data integrity, and transparency. Coming to the fitness of purpose, it's important to define the regulatory agency the data from NAMs are intended for. Either it can be US EPA, EU, or any other country. One has to ensure that the information provided is sufficient to address the regulatory endpoint of interest. While using NAMs, it's important to define the manner in which NAMs are incorporated into the assessment. So that can be a standalone assay or a part of the defined approach or an integrated approach or weight of evidence approach. Also, it is important to define the context in which NAMs are intended to be used, like pre-regulatory screening or prioritization, chemical grouping, hazard identification, or quantitative risk assessment purpose. Coming to the human biological relevance. So here one need to demonstrate that the similarities between the biology that was measured by the method to that of human biology. So when appropriate comparisons are made, it's important to demonstrate the NAMs reflects the human biological understanding equal or better than the traditional animal test methods. One need to evaluate the protocol, equipment use, computational models used for endpoint prediction. One need to assess and then describe the intra-laboratory reproducibility, associated reference chemicals used, controls used, limit of detection, and then quantification as well. One need to demonstrate the integrity and credibility of the data submitted for assessment and for peer review as well. It can be like all the way from raw data to the final report. It's important to assess and then describe the uncertainties and limitations also associated with the NAM methodologies. It's again, it's important to determine the appropriate level of external review necessary. The peer review, publication, human biological relevance, fitness for purpose are encouraged for publications. So in certain cases, especially when novel methods are used, NAMs may be reviewed by the independent third parties. So for industry, we try to ask question ourselves while adopting or prioritizing NAMs for discovery, development, or regulatory purposes. So which may be the same questions that regulators also may have in their mind. So the first question is the sustainable technology profile. So this sustainable technology profile will help us earlier on to remove any unacceptable risk for human health and environment. The second question is healthy R&D investment strategy. So is there a market need for this new solution? Is this technology registrable and suitable for long term? So this means to say that the molecule development will take 10 years. So it's a 10-year journey. So one need to ensure that the methods are safe by design, they are robust, effective, viable from the discovery or development or from the regulatory context as well. So based on our own experience from the analog prioritization context, NAMs are good at toxicodynamics. So here the high to medium throughput screen will help us to exclude certain unwanted pathway activation. NAMs are good at toxicokinetics, wherein help us to exclude the unwanted kinetic characteristics. They are good at general bioactivity, which help us to prioritize analogs with favorable biological activity. See, NAMs are not generally good at animal outcomes to predict intact animal outcomes. So irrespective of the human relevancy or not, NAMs are not good at classification and labeling based apical effects with few exceptions like gene tox or a few acute studies. So let's switch gears a bit towards mammalian toxicity testing of agrochemicals. So at present, the agrochemicals are the most rigorously tested group of chemicals in the market for regulatory submissions. So mammalian toxicology is an important element in this assessment. So some of the core endpoints of mammalian toxicity assessment for registration are ADME studies, acute toxicity studies, subacute, subchronic studies, chronic toxicity studies, carcinogenicity studies, gene tox, DART studies, immunotoxicity, and neurotoxicity. So approximately 18,000 animals are being used for these testing requirements across the disciplines, and that is based on the common guidelines per active ingredient, and approximately 7,000 to 9,000 animals being mammals here. If we consider the acute studies specifically, global regulatory agencies require acute toxicity assessment on every plant protection product. So approximately 50 to 70 mammals are used for single new product. That includes a 6-pack studies, and those studies include acute oral toxicity, dermal toxicity, inhalation, eye irritation, skin irritation, and sensitization. So hundreds of new products are brought into the market or registered globally, and one can imagine the number of animals used for this purpose. See, the basic purpose of plant protection products acute toxicity testing is to evaluate the hazard for classification, labeling, and for worker safety. So plant protection products or formulations may have differing acute toxicity potential compared to that of their AI, because there may be a physical properties that may be different compared to AI, like granular formation, which may cause eye irritation, route of exposure. So dust formation may lead to inhalation, exposure, and then toxicity. The co-formulants within the formulation would also add to the toxicity profile of the active ingredient. So at present, the regulatory acceptance of NAMs within the acute toxicity space is very subjective globally. However, we have options for waiver of in vivo tests. But looking at the trend, it is certain that the ag industry is going through a transition stage from in vivo to in vitro. But at present, we are ending up in doing both in vivo as well as in vitro studies. So based on our own experience, there is an increased trend in acceptability of NAMs for agrochemical formulations. So in general, EU, Australia, and then New Zealand, they accept in vitro tests. US is in a transition stage, but they encourage submission of NAMs data for registration purpose. In general, Latin American countries and APAC countries, they rely on in vivo tests. However, Japan and then Brazil, they encourage use of alternatives. India has a mention of in vitro alternatives as an opportunity for waiver as well. So there have been substantial investments made into the both development of in vitro methods as well as validation of those methods to predict the eye irritation response as an alternative to the classical rabbit drives method. So despite these effects, no standalone in vitro test has successfully been validated to fully replace the drives irritation test for regulatory purposes. So recently, industry made an effort in collaboration with CropLife Brazil, wherein the retrospective data analysis was done for eye irritation in vivo and in vitro test data. For this project, working group members included crop protection companies, and the goal of this project was to conduct a retrospective analysis of in vitro eye irritation assay results from GHS additivity calculation for pesticide formulations compared to in vivo GHS regulatory classifications, and to propose testing strategies for eye irritation evaluation. So 8 companies submitted data with a total of 187 formulations with at least 1 in vivo test, 1 in vitro test, and a GHS additivity calculations. So performance of each was evaluated for concordance, sensitivity, specificity, false positivity, and negative or positive predictivity as well. So here is the result of data analysis. So I would like to highlight the one outcome for today's purpose, that is the negative predictivity. So negative predictivity for epiacular is 100%. It is 86% for BCOP. It is 88 for ICE and 93 for calculation method. So this means that the 4 alternative methods correctly identifies the agrochemical formulations that do not require classification for eye irritation. So there is a high confidence that the prediction of not classified from alternative method is a true not classified for all 4 approaches. So let's look into an example here as an alternative testing approach based on these results. So there are 2 steps here. Step 1, which is an initial consideration, and step 2 is a in vitro eye assessment. So in step 1, assess the potential of the formulation or test material for irritation and then corrosion. So those important points that may include eye irritation or severe irritation data or extreme pH, skin corrosivity, or information on any of the co-formulates. So if the answer is yes, then revisit the whole formulation strategy. If the answer to the first step is no, then proceed to the step 2 and then conduct the in vitro testing, the available in vitro methods, and 1 or more approaches can be used depending on the applicability or with the information availability or compatibility with the test system. So there are 3 possible outcomes here. So if the outcome is negative, combine the information that has been gathered from the step 1 and approach the regulator for a possible waiver. If the result is positive, then again go back to the formulation strategy and then revisit the formulation. So in if the result is borderline, as a last resort, conduct the in vivo animal test. So with this alternative approach, in 2 of the 3 outcomes, use of animals can be avoided, particularly for label and hazard communication purpose. So GHS additive approach. So every attempt is being made to develop non-animal methods to predict acute toxicity. So not many promising or widely accepted replacements available for oral, dermal, or inhalation acute toxicity assays. So basically these 3 acute toxicity assays are based on LD50 RR lethality, and in the past rarely effort was made to determine the mechanism of toxicity. So we don't have mechanistic data available here. So one of the option could be here is GHS additive formula for evaluating the agrochemical for these acute toxicity endpoints. So this GHS additive formula is a computation method. Example, it uses LD50 values for calculating acute toxicity of mixtures based on all components of a formulation. And it is based on the assumption of additivity that the chemicals that are not interactive. This approach is already a part of UNGHS guidance as well as global transport regulation. So ICA uses this approach. US EPA has not implemented the GHS additivity formula for agricultural formulations. However, US EPA encourages to submit the data on additivity. So this GHS additive formula method could be of great use towards 3R, especially for labeling and hazard communication purpose when it is used appropriately. For example, the predictions is better with formulations with low toxicity profile or formulation that doesn't require classification, that is NC. The prediction is better when it is used while accounting for the formulation environment and the existing informations like in vivo data read across to the similar formulations. So I have listed a couple of publications here for those who are interested in additional reading. That's all I have, and thanks for your time.
Thanks, Pradeep. Wonderful. Pradeep. Just a quick question I have. You've given the threshold acceptabilities. I mean, it's 60%, 68%, 80%, 90%. In your personal view, what should be the acceptable percentage threshold acceptability limit that you may, in your personal opinion, may like the regulators to consider?
Yeah, it all depends. It all depends on the case by case basis. So essentially, like if we have the information that is towards the MC, then probably we can consider and then proceed with that.
Thanks, Pradeep.
Thank you.
You need to.
Yeah. Thank you, sir, for the presentation. Now I request Dr. Ankita Pandey, ma'am, to present Dr. Pradeep Kumar, sir, with a memento. Thank you, ma'am.
Okay. Thanks, Pradeep. Ladies and gentlemen, may I request Dr. S.K. Jain also to join for the panel discussion. Pradeep, you're also go there. I'll also like to introduce another panelist that we would very likely who's here in the audience and is someone whom we can look upon for further guidance in this regard. May I request Dr. Debrata Kango, sir, to please come on stage and preside over as a panelist, sir, please. Dr. Debrata Kango, just a brief introduction for those who may not have known about him. He is an MBBS and an MD in pharmacology, subsequently trained in advanced toxicology and human health risk assessment in RVIM, the Netherlands. Further training in health hazards due to modernization of agriculture in Japan. Retired as an additional Director General, Ministry of Health and Family Welfare, Government of India. He has 41 years of work experience in regulatory toxicology, dealing with human risk assessment in national and international level. At present, chairperson of the Research Advisory Council of CSIR IITR Lucknow in India. Formerly member expert medical toxicologist with the Central Insecticide Board and Registration Committee, Government of India. May I request my co-chair to kindly start the panel discussion this regard. Over to you, Ankita.
Thank you, Mr. Saini, and thank you to all the speakers today for delivering such an informative and insightful presentation. We have a couple of questions we'll try and get to in the next 20 minutes or so, since we are running a little bit short in time. Dr. Jain, the first question is for you. I mean, we're seeing that CIBRC has been accepting many of these approaches since 2017, right, as per the guidelines. And we know that a lot of, but like you said, that we are not seeing a lot of in vitro submissions in India. Mostly you're seeing waivers based on weight of evidence approach. So as I understand, many global companies have been submitting a lot of alternative data in Europe and USA. And even though Indian regulators are accepting it, still there is certain hesitance to submit it in India. So why do you think there is this disconnect, even though the regulators are encouraging, but the industry is still not confident to submit? So it's basically a regulatory implementation point of view.
We can't say why applicants are not submitting the international data, particularly in case of the import cases. They are importing the product from that country, so they can use that data if they have on names or other ones, can be considered.
Dr. Pradeep, do you want to add more insights into this?
So, add it here. So basically, the NAMS is a evolving science, and important is that it is based on fit for purpose. So there are a lot of methods are being developed globally, but we are not in a same speed for validation than the development. So we have limitations of the methods that are both methods as well as validated methods. So that may be the one reason, maybe that's India or global. The second thing is that, like, from the industry perspective is lack of harmonization in terms of acceptability around the world. So with that, as an industry, we end up doing both in vivo and in vitro results, and still we will have the luxury of in vivo animal experimentation methods as well. The other thing is that, like, the in for validation, the data sharing. So that's another area that we can improve on and then prioritize and then focus on the need of the hour. So that will help in developing and validating more and more alternatives, and then we submit the data to the regulators.
Thank you. Dr. Kanungo, do you have any thoughts on how we can address the situation?
Am I audible? I think let me thank you for inviting me. I thought that I will be an audience, but you dragged me here. Anyway, let me tell you one story or whatever from my childhood. Our teacher used to give a paragraph or essay on cow. So you have to, whatever the teacher says, you get by heart and you have to written. Even if you know certain other sentence, you can't write. Numbers will be maybe deducted. So here the problem, whatever you ask question, it is the mindset of both the registrant as well as the regulators. Neither the regulators have changed their mindset, nor the registrants have changed their mindset. Registrants have the data. Why would they fear they don't submit that? Even if they are submitted, they will be asked 101 questions. This is first thing. Both registration, I mean the regulated and the regulators have to change their mindset that yes, I will go for the other things. Any other question? Maybe in the second round I will come something.
Okay. Mr. Saini, do you have any thoughts on how we can bridge this gap?
Yeah, well, I do agree with Dr. Congo. It's a matter of mindset going across and accessibility. And it is also a challenge for all of you as students, academicians, also to take into account the current regulatory requirements so that your research is oriented towards that kind of acceptability within the regulatory framework. So that's my take on the whole issue.
I'd just like to add something here. I mean, of course, we need regulators to see more data to have that confidence, and we need the industry to submit that data. So I think one of the ways that we can address this is to have maybe say pre-submission consultation. So we don't have like a formal channel of doing that in India. So in other countries like US and EU, they have like a formal process in place where the industry gets in touch with the regulators before they start generating all the data. And instead of taking like a checkbox approach, they actually discuss what is really needed to, inform or make health protective risk assessments. So this helps in moving away from checkbox approach, and also it gives an insight or a little bit of confidence from the regulators to say, yes, in this situation, an in vitro method or a read across or an in silico method may work. So I think that could also be one of the ways by which we can, having like more scientific dialogue to clarify data needs and applicability of a particular NAM. That could be one of the ways. That's a solution.
Huh?
That's a solution that I was giving to address this issue.
Okay, let me tell some of my experience. I have got international exposure to various regulators throughout the globe. Whenever any question comes, any product comes, on the basis of my experience, I am saying there's a need to have I need to have what I want. My responsibility as a regulator to protect the human health, whatever the exposed from, whatever either from inhalation route or oral route or the dermal route, whatever it may. So in that case, what happens? I have to, maybe along with the registrants or the whoever coming with the application, both sit together and along with some peer reviewer or some things, whatever we want, we first look into the compound, maybe in silico, then see what are the general requirement and what we want, exactly what we want for the registration point of view. The second one, having said so, maybe on the in silico point of view or structure activity relationship, the chemistry we need to, as a toxicologist or regulator, we need to have the chemistry knowledge something. Having said so, what we will do is, are we flexible? The most of the regulators, those who are there, they feel two things. There are guidelines. There is a checklist system. So they follow the checklist. They don't have, they little secky not to follow or to follow the checklist or not to follow the checklist. They should have a flexibility. There are certain countries, it is noted, all the guidelines are noted in their act and rules. There is no flexibility there. But in our country, the guidelines are not a part of the rules and act. So the regulators have a lot of flexibility. Then having said so, that I have flexibility and I know what is my point of departure, I have to find out what is my end point. Then I will come to the weight of evidence, whether I have got sufficient evidence to find out. If the sufficient evidence is not there, then I have to find out which one I want more. If that is, then I will come to the question of NAM. Whether there is any NAM is available, either in silico or in chemico or structure activity relationship or bridging gap or any one of the study already given, from that I can find out. I'll just give one example. Abhay was telling about URG, extended one generation reproductive study. If you have that study, you don't need to have immunotoxicity. We don't need to have neurotoxicity sometimes, and you don't have the developmental toxicity in rat. So this is what this particular one study will give you 4 parameters instead of, because in checklist it is there, you have to go for immunotoxicity. You undertake immunotoxicity, or if a checklist says that you have to go for rat developmental toxicity study, do it. So all these things you need to it. Then in that NAM, you can find out, tell it is number 3. You just note down 1, 2, 3, 4. These are the NAM or novel alternative, whatever it may be. NAM, some say, some people are saying it is a novel animal study, a newer animal study, novel alternative. Methodology. In that methodology, I am getting lot of weight of evidence to fulfill my desired things, so there is no need. If at all something known, suppose 1, 2, 3, 4 studies again I want, then I have to find out whether those studies are having any NAM. Either animal studies or non-animal studies or computation methodology, all these things. Like there are certain DNT studies, developmental neurotoxicity. You can have alternative study. Acute toxicity, you can have bridging concept. So there are so many studies for which the NAM is concerned. I will not say that it is only the non-animal studies, but different studies. I'll give you an example of one particular chemical, 1,4-DMN. Recently WHO was reviewing, I was the reviewer last September. there are certain metabolites in that particular, this particularly, please don't quote that name of the product. There are certain metabolites which are coming in the food or the crop on which it will be. So ideally, the applicant or the resistant need to generate data on those particular. So what we did QSAR analysis. As Dr. Jain was saying, the QSAR analysis, there are several type of analysis, QSAR, Nexus, Derrick, OSIS, all these several things. And everybody's endpoint is different. Something is statistical based, somebody is biological based. So you combine together and find out whether it is genotoxic or not. Then take a decision based on the TTC, threshold of toxicological concentration. So this is called NAM. NAM is you have to see to approve a product on the basis of the weight of evidence without asking for further data. Thank you very much.
Thank you, Dr. Tango. I think you made a very strong point here in terms of having more regulatory flexibility. These guidelines, sorry, Dr. Jain, please.
In the revised guidelines, that is 2023 guidelines, we have already included this provision that resistants, they are free, they can fully discuss and then start generating data.
Problem which I'm saying. You see, science is a thing. I don't think single-handedly one can understand everything and we can do one thing. One-to-one discussion with a regulator, one regulator with one regulated. Regulated is always under lower hand. Whatever regulator will say, yes, sir, yes, sir, yes, sir, yes, sir. It is always better to have a peer review, always better to have even WHO, even EPA, EU. They have a group of toxicologists who discuss to find out whether there is weight of evidence is there or not. Here in our country, we do not have. It's always a single-handed, including me when I was there. If I am doing a mistake, nobody is there to correct me. With this gray hair now, I am very open to tell, but this is what is going on. There is a need to have some peer review system. Otherwise, it will be very difficult. Thank you.
Thank you, Dr. Tango. Also, when we're talking about the regulatory framework, I think as and when we see new technologies evolving, the regulatory framework needs to keep pace with these changing technologies. So how can we ensure that, whenever we have new methods, they are fully incorporated within the regulatory framework? Can we have like a channel or an opportunity to have these regular discussions with the regulators on a maybe 6 months or a yearly basis to discuss what these new methods are, what is the applicability for regulatory purposes, how can we incorporate that within the regulatory framework? Dr. Jain.
This area is open and we have options for it.
From the industry perspective, we will be very happy to bring our experience elsewhere in the world onto the table. So,
could we go to the government with this request to have maybe say an expert committee set up to review these NAMs, or can we expect like a yearly stakeholder discussion with the regulators, companies, experts all together reviewing NAMs on a regular basis to ensure that we are at pace with what is happening internationally? So basically, it's about accelerating regulatory acceptance of new methods.
Let me tell, this constituting expert committee is not going to help unless the mindset of both regulator and regulated are changed. Number one, the use of the norm will vary from product to product, structure to structure, study to study. So no general category recommendation will help. Am I right, Dr. Gupta?
This is a very interesting and progressive discussion, and I fully agree with what Ankita said, that we have to basically, as a society or as an expert or as a key opinion leaders, we have to make a case that yes, see, these are the amps which are available, which are validated. And as I said in the morning, there are 2-3 categories. One which have been validated and which have been accepted outside, maybe OECD. So there's the easiest low-hanging fruit where we can say, yes, why you don't accept it. And the other is which is being validated are still in the process of global validation or in the country. So that is where you can say, yes, these are the process which are being validated. Will you be accepting this if this is comes to you after 4 or 3 years? So that we get ready for that. And the third is in the infancy stage, here there is not validated, but people have started thinking about it. Maybe IITR, JRF and other people are working on it. Involve them right from the beginning that would you be willing to accept such in future? So I think what you said, and you said that this interaction between the regulator and the scientists like this and such type of forum will be of great value. The other thing, like in clinical trial, there has been concept which, in fact, I was the chairman of drafting committee. We started the pre-IND consultation. They have to pay 5 lakh rupees, the industry, and have a pre-IND consultation in which the protocol is given to them, and then there is a free frank discussion. So there can be sort of a thing like that can also be suggested to them. Yes, this is the PCT, which is preclinical toxicology pro forma, is submitted to them, and then say, yes, is this acceptable or not? So that when we submit, there are 2 ways. One, you submit the RAM, and then it is up to them whether they accept it or not. When we talk of a mindset training, the other thing is you submit the protocol, ask them whether this will be accepted, so that we do not waste our energy and time. But I fully agree with Dr. Kanungan. I also agree with Ankita. We have to make, this is the purpose of this. I think this can be taken up by society like this.
Thank you, Dr. Gupta. I think in some cases we are really stuck in the first step where we have validated and accepted methods, but still CIBRC is not very receptive. For example, the GHS mixture equation.
These preclinical things. We can also, in case of pesticide, we can also suggest for the pre-submission consultation. So that, that's what I go to the point number 1, that we have to have a pre-submission consultation to find out where do we stand, what do we want. A product which is not, for example, which is from the ADME study, we found that there is no absorption. Why do you require the bioavailability or other studies? So it is not absorbed in the GI tract. So this is what I want. There should be a consultation. Thank you.
Thank you very much, gentlemen. And Ankita, lots thanks to you. I just have one question to the remaining panelists, Dr. Jan and Dr. Pradeep. Just a quick thing. We've spoken about earlier, Ankita told us about the roadmap on NAM in the US and the EP. And Dr. Jan has also given us an insight that they are partially acceptable and the progress is being made across. Just a quick question, one-liner. Do you see a difference on these NAMs with respect to chemicals, botanicals, and microbial agri inputs? A quick, the first of you.
Yeah, it's fundamentally it's for fit for purpose. We need to look into each and every aspect.
No, are there depending upon the property?
Probably,
depending upon the property, like microbials have to. Do you see any kind of challenges coming across?
Exactly, we need to look into the fit for purpose.
Right, Dr. Kango, you may. Dr. Jan is on the phone, I believe. Dr. Jan, any take on this? Do you find any challenges in such kind of NAMs with respect to chemicals? We generally talk about chemicals, but what about in the case of botanicals and microbes?
we have already included the fire, including technicals, and we'll be looking case to case basis that can be considered.
Right. Dr. Congo, based on your international experience, would you like to have something?
I'm not wrong.
Yeah.
You're asking whether the concept, I mean, the methodology will change from one to one to one.
Exactly.
So what is NAM? NAM is a concept.
Okay.
So how can that concept will change? As Dr. Jain told, it will be on case to case basis. You have to see NAM is there in our mind. And we have to look into this case, irrespective of the chemical, irrespective of the biopesticide, irrespective of the things. Then we'll go these 4 stages which I have told. Pre-consultation, find out what is the requirement. Second, whether I have the flexibility to work for it. Third, what are the NAM already available? Fourth, what are the NAM I want to have it? These 4 steps we have to do as a regulator, as a regulated also.
Thank you very much, Dr. Congo. Ladies and gentlemen, with this we end the panel discussion, and you've been a wonderful audience. And I thank my fellow panelists for wonderful interaction, especially the one Dr. YK Gupta also rose up to the occasion. Thank you very much, sir, for pitching in. And once again, this is Vipin Saini and Ankita signing off for the day. Thank you.
Devabrata Kanungo, sir, to accept our memento as a token of appreciation. I request YK Gupta, sir, to present Devabrata, sir.
What an honor. Huh?
Thank you, sir. I request SK Jain, sir, to present Ankita Pandey, ma'am, with a memento. Thank you, sir. Now I request Dr. Devabrata, sir, to present Mr. Vipin Saini, sir, with a memento. Thank you, sir. Before the next session, refreshments have been arranged outside, so please join us for the tea break. We will resume the session after 15 minutes.
Yeah.
Welcome back, everyone. For the next session, I request Professor Y.K. Gupta sir, President, Society for Alternatives to Animal Experiment, to be the chairperson, and I request him to introduce the speakers. The first speaker of the event will be Dr. Rohit Bish sir, who is a PhD in ophthalmology from the University of Auckland, and he is a senior scientist. He has done various research projects in AIMS and University of Missouri, Kansas, and his work has been published in many high impact journals and presented at many international conferences. I request him to come over the stage. The next speaker would be Dr. Ashwin Kumar Bavarkar sir. He is serving as the senior expert in the APAC field marketing at Merck Life Sciences, and he is proficient in the use of advanced PCR methods and has successfully managed rapid food pathogen technique. I request Ashwin Kumar sir to come over the stage. And the next speaker would be Alden Fernandez sir, the head of operations at Genie Laboratories. He has been pivotal in many diverse teams, ensuring implementation of technologies and making R&D safer and sustainable. I request Alden Fernandez to come over the stage. And the last speaker would be Dr. Kalaivani ma'am, Senior Scientific Officer, Biologics Section, India Pharmacopoeia Commission, who will be available online for this session. I request Dr. Rohit Bisher to commence his presentation.
Okay. Thank you. Okay, fine. Good evening, everyone. First of all, I would like to thank Society of Alternatives of Animal Testing India for organizing such a wonderful and much needed platform where this professionals from diverse groups coming together, interact and give ideas, which is very helpful for organization like us to prepare a roadmap to achieve our common goal, that is advancement alternative to animal testing. And I would also like to thank for giving this opportunity, a session to share the information regarding the recent updates on alternative to animal testings in pharmaceutical sector. And not the least, I would like to thank Aligarh Muslim University. A lot of conferences going on. It's very difficult to, manage each and everything, but they are doing their best to make this conference a big success. So thank you very much. Myself, Dr. Rohit Best, I am a scientist at Regulatory Toxicology Department, PETA, India. I work with regulators, policymakers, scientists, and stakeholder companies for the advancement of alternative animal testing methods and for the replacement of use of animals, which are used for the production of human therapeutic products with new technologies like recombinant technology. Specifically, we are currently working on monoclonal antibodies for that. So today, my presentation is focused on some recent advancement and alternative animal testing in pharma sector. I will share some information about animal testings. What are the limitations with the animal testings? What are the different type of alternatives so animal testings are available? Some global scenario and what new recent advancements is going on these alternative animal testing development in India. Then we'll focus about on pharma sectors. In the previous sections, my colleague Dr. Ankita already discussed about the alternatives testing in agrochemical and pesticides sectors. So in these sections, we are focused on the Indian pharma sectors. So what type of initiatives PETA are doing for the advancement of alternative testing methods? And then, of course, we will discuss about the challenge and opportunities for the advancement of these alternative testing methods in India. So first of all, the limitations with the animal testing methods. According to this US National Institute of Health, the world's largest funder for the biomedical research, the failure rates of the drugs is about 90%. And it also stated in the various reviews, meta reviews, that 90% of drugs fails, which show a very promising results in the preclinical testing, fails ultimately in the human clinical trials. And an investigation report in 2015 also conclude that about 89% of the preclinical studies based on animal test methods could not be reproducible. So reproducibility is also a very core problem with this animal test models. And of course, the development, the discovery, it takes a lot of time, a lot of money, and animal ethics is also involved. So it's not about the ethics only, it's about the financial burden. It's also about saving the time so that the drug will cross all the hurdle and available for the patient in the market as soon as possible. So what are the different type of alternative animal experiments or models are available? So this is a new reports on non-animal alternative test market size. It's a global opportunity analysis and industry forecast for 2021 to 2030. So this report forecasts about alternative testing methods into different sections like biotechnology, which can be a cell culture, high through output methods, molecular imaging, omics technologies, by methods like cellular assay, biochemical assays, in silico and ex vivo models, by end users like, of course, pharma industry, cosmetics, household products, diagnostics, chemical industries, and food industry. So this report shows that the market value of alternative testing methods in 2022 is about $1.7 billion. And it is forecast that its value in 2032 will be about 4.5 billion, where the major chunk will be of organ on chip segment and ex vivo segment, then cell culture technology and infectious disease segments. And this overall 653.8 million, it's only the market share at North American market values. So if we see the market size, the growth, which is divided by the end technologies. So we see that the cellular cultures, these are all the sections, including comparatively cell cultures models are at high. Definitely at biotechnology, cellular assays will be more used, development will be at faster rate in 2032. And at the end users, of course, it is very low as 2021, but it is expected that it will be highly increased in 2032. So when we talk about alternative testing methods, there are different type of models available. We divided it as a three-dimensional tissue models, in which we have reconstructed human tissue models. We have spheroids, we have organoids, we have precision cuts organ slices, and we have bioprinting. Then next segment is microphysiological systems, that is organ on chip, We have different type of microphysiological system, and a lot of research is going on in these organ chip segments. Even in India also, Academic Research Institute has initiated a lot of research in it, and now they are also interacting with the regulatory bodies to get into the pipeline. Next is the cell-based assays, and next is in silico models. So what we have experienced with in silico models is sometimes the regulators are, hesitate to accept in silico model as alone, but nowadays the couple model system is also under the way, where the in silico models are coupled with in vitro models for the toxicity analysis. So at the global scenario, there are different type of models which has already been developed. Major player is like MedTech USA, which is already developed epiocular models for eye-based irritation testing, and this model is already validated and integrated on OECD test guidelines. Skin Ethics, L'Oréal France. This is for skin irritation testings, EpiSkin. So there are different type of models which are already validated and integrated in the OECD test guidelines. So as initially we have discussed by OECD test guidelines, but the major hurdle is that when we focus about the alternative testing method, the majority of these models are already tested on agrochemicals and chemicals. But when we discuss about the pharmaceuticals, whether it's as an ingredient or as the final products, you will find a very low number of alternative models which are tested on pharmaceutical products. So this is a hot topic of discussions which is going on, like whether we can use this system as such and test for the pharmaceutical ingredient compositions and as a final formulations, or we need to do modification in this method to suitable for the pharmaceutical formulations. We have recently do a collaborations with a company in India, that is Biodimension Technology. It's a VIT incubator based company. Now they are in Bangalore also. These companies working on 3D tissue models. They have already validated their 3D tissue models for skin. Now we are working with them for the OECD integration system, and we are in communication with DST also for this. And we are, as per the communications with the DST, we are received very positive response. So the Indian global regulatory bodies are also, understanding the value of these alternative test models, and they are very acceptive, and they are having a positive response for the approaches made by the industry for this alternative testing method. For organ chips, there are different type of organ chip models now available, and it is projected that this market sizes also increase in 2028. Next is PETA initiatives. Now, what PETA is doing for the advancement of alternative to test methods, there are the two important bodies, regulators, and of course the stakeholder companies. This change will be happen, the combination of both. But we are the team, we work with regulators, policymakers, stakeholder companies. We sometimes work as a bridge for the communications. Sometimes we propose technical comments and proposals where there is a possibility of replacement of the animal test methods with the replacement of alternative test method. We generally highlight those with the regulators and the stakeholder companies, and we also interact with the stakeholder companies, the restrictions or the problems they face, which restrict them to make this such type of transition. So we do meeting with regulatory bodies and policymakers. We do meeting with academic scientists. What many research work is with the academic collaborations. These type of initial models are developed in academic science, and then we do a collaboration with the industry to further scale up and to make it develop in a industry style. That is very different when lab scale models is scale up to an industrial setup. We do meeting with the stakeholder companies. We do meeting with government testing laboratories. We do R&D collaborations with the academic scientists and industries. Time to time, we submit technical comments and proposals for the areas where there is a need of replacement animals with the alternative test methods. We provide research and travel grants to promote the alternative test methods in India and abroad. We provide hands-on training opportunities for early career scientists and senior scientists. We provide guidance to the industry for regulatory work. just I have mentioned like this biodimension technologies. We are working with them for the integrating their three-dimensional tissue models in OECD test guidelines. And we participate in the conference workshops to promote and share the information with the individuals regarding this alternative test method information. So our ongoing projects. So here I will highlight some of our projects which is ongoing. First one is the animal-based pyrogen testing methods. Initially, if somebody knows about this pyrogen test method, it is done on rabbit, that is rabbit pyrogen test method. Now, after long communication with Indian Pharmacopoeia Commission, pyrogen test from Indian Pharmacopoeia, and it is replaced with bacteria endotoxin. So now we are communicating with the Indian Pharmacopoeia to remove this bacteria endotoxin test with monocyte activation test and recombinant
इंटरनेशनल कॉन्फ्रेंस है एक सर्टिफिकेट
So we have
इंटरनेशनल
achieved
मिल
so much
जाएगा कि
success
इंटरनेशनल
from
कॉन्फ्रेंस
long time now. We are at the end stage where the regulatory body is asking for the validation data to make such changes in the Indian Pharmacopoeia to include these alternative test method in the
एक्चुअली क्या होता है
initially
ये
with the
ऑफलाइन तो
vaccine
करते हैं
monographs.
लास्ट में ऑनलाइन ओपन कर देते हैं तो कई सारे
Second,
लोग
we
ऑनलाइन
are working with the non-animal based therapeutic
तो
products
उनका
like
एक्स्ट्रा
recombinant
वो
monoclonal
ऑनलाइन में पैसे
antibodies.
तो कुछ लगने नहीं है 2-2
Initially,
ढाई
these
रुपए
antibodies are produced using plants. We do the
भाई
hyperimmunization of these process, and then we collect the blood and serum, and then we produce these antibodies. But this is a very painful, and it has a lot of limitations. Like it is having a risk of serum sickness, hypersensitivity. The serum is having a very short half-life. Batch-to-batch variability is there. So the best options for this is to use the advanced technologies like recombinant technology. So we are in communication with DBT and BIRAG to promote this development monoclonal antibodies with using recombinant technologies. So next is our new non-animal preclinical safety testing strategy for pharmaceuticals. PETA scientists have developed some monoclonal antibodies for antivenoms and some other diseases. So we have till now we have developed more than 400 monoclonal antibodies. We are into research also. So we do research with the partnership with the academic and industrial research facilities collaborations. So in this, we have developed a new safety testing strategies for monoclonal antibodies that are directed against specific foreign targets. So we have communicated this with DBT BIRAG also, and now we are at the stage where we are submitting a full proposal to execute this, to validate this test method in India in collaboration with the industry. Next is animal based test method for vaccines. We are working with the Indian Pharmacopoeia and drug testing laboratories. We are also working with the National Institute of Biologicals to replace the possible places where there is a possibility we can replace the animal-based test method with alternatives like mass neutralization test. We in this mass neutralization test, this test for the vaccine is not used in anywhere in the world. But India, it is still in use, and it can be replaced with the RFT or ELISA. So we are in communications with the Center Drug Testing Laboratories and IPC, and we have reached that place. That soon it will be these alternative methods will be included in the monograph. Next is animal-based test requirements for biosimilars in India. Recently, we have a meeting with DBT where this animal-based test requirement for biosimilars, it is not required in any countries, any global regulatory bodies, but India still be required this animal-based safety testing, preclinical safety testing methods for the approval of biosimilars. So now this DBT have accepted our proposal. We have in collaboration, we have initiated this project in collaboration with different biosimilar developers of India. Now they have in communications with the stakeholder companies, and soon it will be this requirement will be waiver for the biosimilar developers of India. So this is the PETA India research grants, which every year we provide the grants for early career scientists or the pharmaceutical companies which are working on the alternative testing methods. So to 2023, this grant is we have granted this to Biodimension Technologies, which is working on 3-dimensional tissue models, and in that their skin tissue models already validated, and now they are moving forward for OECD test integration. Next is we provide the hand-on training travel grants. So last year, so this is in collaboration with the Institute of In Vitro Sciences. So we select the student, early career scientists from India and abroad for this hands-on training experience, which will be very helpful for them for their PhD or postdocs for the advancement of this alternative test method development. So this is another award for 3D human tissue models alveolar macrophages to assess the toxicity or drug efficacy in respiratory tracts. So we are, one of our areas also this epithelium toxic inhalation toxicity. So this is the grant which is come under that category. There are different type of research award in different categories. These are some highlights like 3D tissue model, recombinant antibody challenges grant, inhalation toxicity. So these are the different ways we promote the development of alternative testing models. And we encourages the early career scientists or researchers from diverse area to apply for these grants. So what we have achieved so far in alternative testing methods. So there are some highlights, like we have proposed submitted one proposal to Indian Pharmacopoeia Commissions to have an expert working group committee for alternative to animal testing. Now IPC has accepted it, and it is now on official and already conducted 2 meetings. So we have one committee in IPC for the dedicated towards this alternative testing methods. Second, complete removal abnormal toxic test from Indian Pharmacopoeia. Now this test is completely removed. Rabbit pyrogen test is almost completely removed from IPCA and replaced with the bacterial endotoxin test. And now we are working to remove it, replace it with monocyte activation test and recombinant factor C. And the replacement of mass neutralization test for vaccine with Rift and ELISA, and animal safety test requirement for biosimilars. So I have already discussed these type of areas which are under communication or under progress with regulatory bodies. Next, this is the most important thing with the alternative testing method, which need time for discussion, that is challenges and opportunities. The first biggest challenge is the cost. The most of the alternative test methods which are already validated or integrated in the OCD test guidelines or other regulatory bodies which are accepted, not only FDA but other, they are mostly developed in the Western countries, and the cost is very high. So it is always better to initiate a Make in India, and more startup companies should come, like Birak. Now they are, funding, and they are helping us supporting more and more startup companies. So like biodimension technologies, other companies are also coming up, making these type of alternative test models in India, which in futures surely make this the cost of this alternative test models very less. Second is the lack of hand-on training experience, that when during the communication with the stakeholder companies, this is also a major problem when there is a lack of facilities and there is a lack of hand-on experience. Now the regulatory requirements. So like in one project, when we approach for this alternative test models, which is already been validated, which are existing in the market, there are a lot of documents already existed in the internet, and there are a lot of validation documents is already available and conducted by the pharmaceutical industry. But if we want acceptance of these alternative test models in India, we have to conduct these validation studies in the Indian drug manufacturers, and then there will be a discussion within the regulatory bodies how to whether they to accept or not these validation data. Second, funding opportunities. We are in communication. There is as already mentioned during these sessions, that there is very less funding opportunity for alternative to animal testing methods. But we are in communication with to DBT, BIRAC, and DST particularly. They provide a lot of research funding, but we are having also preparing a proposal to to give a small chunk of, at least as a initial step, start with a small chunk of funding for dedicated towards the toxicological study, particularly focus on the development of alternative testing methods. And this type of funding opportunities for alternative also promotes the R&D, and then these have many places of new companies or academic research professional will also come up with the new models. Now, this method validations is also a problem with the acceptance of these models in India. What type of validation study, what type of validation long-term, short-term, what type of parameters these type of uncertain, these are uncertain, unclear. So there is a regular communications needed between the regulator, stakeholder, company, what type of regulatory data they accept. And of course, there's an efficient way to promote these alternatives, to make these alternatives in India, to promote the company, allocate the fund to the startups, and to promote the research and development towards the alternative testing methods. Thank you.
Hello.
So good evening all. So I will not take you through the quick presentation on the monocyte activation test, as Rohit made my job very easy. So some basics I will take you through. I will take you to the pyrogen detection, as Rohit mentioned that rabbit pyrogen test is popular in India, and across the globe it is going to eradicate. In 2010, EP come up with the initiative that non-animal testing is required. So for the alternative to rabbit pyrogen test, there is a test called as a monocyte activation test. So in monocyte activation test, what happens? Exactly, we are mimic the human immunologic reaction. So basically, what is the pyrogen? Pyrogen is nothing but a substance. When it comes into the bloodstream of person, it gives the fever response. So pyrogen is basically contain the endotoxin as well as non-endotoxin substance. So in coming slides, I will explain you what is the endotoxin and what is non-endotoxin substance. So when it react with the human immune system, it forms the cytokines. We all aware that cytokine is a substance. These are the in the form of interleukin beta, interleukin 6, interferon gamma, and tumor necrosis factor TNF. So when it induce, it will cause the fever response into the human body. So pyrogen are the two types. Basically, pyrogen is we all aware that endotoxin pyrogen comes from the lipopolysaccharide from the gram-negative types of microorganism. When it gets killed during the processing of the drug substances, I will talk on this specifically on the drug aspect. So it releases that lipopolysaccharide, and when it comes in contact with the bloodstream of the person, believe me, that drug is going to the one day born infant. So that is the criticality. So as a safety aspect, when it comes in contact with the person's blood system, it forms the fever response. And this fever response is so lethal that person may get died because of the fever. And these are the certain non-endotoxin pyrogen which come during the processing of the drug. For example, rubber particles or chemical dust. or some of the environmental flora which are in the manufacturing environment or from the water system, like gram-negative flora or some of the fungal spore. So that also cause the fever response in the human immune system. So when we need to claim the drug or any injectable drug to be free from pyrogen, so sterile doesn't mean it's a pyrogen free. So that's why pyrogen test is required. And again, industries are going for the endotoxin test. That doesn't mean we are fully, I would say, eradicate or rule out the possibility of the non-endotoxin pyrogen, because in endotoxin test, we are only targeting the lipopolysaccharide from the gram-negative version. But what about the non-endotoxin pyrogen? So only pyrogen test will tell you whether drug is safe to release into the, or can we claim it as a pyrogen free or not. So now across the industry, basically pyrogen test is done with the rabbit. So 3 pair of rabbit, there is a preliminary test and main test is there. So I don't want to go into the detail about the rabbit pyrogen test, because there is an alternative for the rabbit pyrogen test. So what is the monocyte activation test? So monocyte activation test is a very simple test where we are allowing drug product to dissolve in certain level of the concentration, that is called as a concentration limit, CLC. That means the threshold at which pyrogen will give the response. So at below that particular threshold, we are diluting this product and we are targeting this product along with the monocyte activation or artificially prepared or biotechnologically derived cells that we called as a monomax 6 cells. So you can see these types of cell lines are available. So I am just talking about the monocytic cell line that is artificially derived by the biotechnological way, since we are promoting here non-animal testing. So this monomax 6 cells, when it come in contact with the pyrogen, then it forms the cytokines. So here cytokine we are only targeting interleukin 6, and interleukin 6 specific ELISA we are running. If substance is a pyrogenic, it forms the interleukin 6, and this interleukin 6 we are detecting by the simple ELISA method, that is a fluorescence based method, and by the reading into the reader we will conclude that whether product is safe or product is non-pyrogenic or not. So simple test is there. If your product is a pyrogenic, then along with the monomax 6 cells, and we are running the interleukin 6 ELISA. So you can see the test setup is very simple. It's take only 22 hours to 24 hours depending on your workflow. First day you need to prepare the standard concentration of the I would say endotoxins and non-endotoxin pyrogens, and we need to inoculate the cells along with the endotoxin and non-endotoxin pyrogen. Then we are incubating into the CO2 free environment. After 24 hours, we are taking the aliquot to check whether interleukin 6 is produced or not. So specific ELISA plate is there, which is already attached with the antibody specific for the interleukin 6. If your product contain interleukin 6, that antibody tag that interleukin 6 and which form the fluorescence, which is simply detected by the fluorescence reader. And if your threshold is low, below the detection limit, you can conclude that product is pyrogen free. So it's a very simple test and biggest alternative for the rabbit pyrogen test. No exploitation of the any animal here. As earlier mentioned by Dr. Rohit, is certain animal test is having the limitations because of their nature. Sometimes this rabbit are undergone the fear, so that they will intentionally, or I would say they are naturally give the fever response, which is gives you the false indication whether drug is having the pyrogen or not. Sometimes your drug is free from pyrogen, but due to the natural fear of rabbit, which gives the pyrogenic response. So that is a false positive indication. So biggest alternative is a monocyte activation test, and it's a very simple test. You can see the workflow here. If you have any specific question, we will discuss during the panel discussion. Thank you.
We'll have questions at the end. Maybe.
Request Alden Fernandez sir to come and assist.
Yeah. Good evening, everybody. First of all, I would like to thank PETA India, AMU, and the society for giving me this opportunity to speak at this event. Thank you so much. I know it's pretty late, so I'll keep it pretty short and run through some of the slides. So my topic will be on efficient and human use of animals in biological production and the search for the alternatives. So it will cover basically the world of biologicals, the unmet need, manufacturing process so that you know the entire process, how it goes through, the regulatory challenges and guidelines which are governing this, then the smart approach, which is both on the therapeutic, on the quality attributes, and the non-animal based alternatives. So let's say this, animal experimentation should be done only if you are sure that the suffering is minimized. First thing. The second thing is there are definitely gained benefits when you're going to use animals in an experimentation. So, and the another thing is the freedoms. So first, the animal should have freedom from thirst and hunger. They should not have any discomfort, no pain, injury, or disease. Hormonal behavior should be normal, and fear and distress should not be there when you're conducting experiments. Just to give you a brief, there are over 115 million animals which are used worldwide for the production processes and for quality control testing, and major being rats and mice. So this is the entire world of biologicals in a nutshell. So if you talk about initially, it will be animal-based biologicals. You have the anti-thymocyte globulin, which is basically an immunosuppressant drug. It can be produced in an equine model, in a horse, or in a rabbit. Then you have anti-rabies serum, anti-scorpion, anti-snake. All these can be either produced in equines, which is the animal of choice, or it can be camels or sheep. Other products which are done in some parts of the world are anti-digoxin antibodies, which are again done in equines. Then the human biologicals world consists of the hormones, the natural hormones, urinary derived, which is HMG, HCG, FSH, and the plasma products, which are gamma globulins, albumin, factor VIII, and so on. Then you have various vaccines which are used in the industry. To name a few, rabies, influenza, dengue. Monoclonal antibodies are there, which are replacing all the left side antibodies soon. Then you have the cell and gene therapy, which is the new industry which is emerging, and other non-biological molecules such as it is used for anesthesia products, it is used in antibiotics. All these things are animals are used in all these. So let's understand the global scenario. So currently, if you look at the numbers, you have more than 100,000 deaths occurring in Asia alone. Out of this, almost 54,000 deaths occur in India itself. And snakebite is a major health problem in India. Almost more than 3 times this number, they suffer from permanent disabilities. WHO in 2017 declared it as a national neglected tropical disease. And since then, they have been working actively with individual countries to ensure that by 2030, there is complete elimination. Just to give you a brief on what we are going to talk about is snakes can be divided into multiple categories. So the two major type of families you would say is the elapids and the viperids. The elapids being the naja naja or the common cobra, and the Bungarus caerulus, which is also known as a common crate. And then you have the viperidae species, which is Daboia russellii, the Russell's viper, and the Echis carinatus. So the viperidae family mostly has high molecular weight toxins, which are big in size, and they are more towards hemorrhagic and coagulopathies. Whereas the elapids have smaller molecular weight toxins, and they are more focused towards neurotoxicity. They will be different types. There will be your phospholipases, your three-finger toxins, serum proteases, serine proteases, L-amino oxidases, your myotoxins. All these proteins can make an impact. Now, to develop antibodies for this, it's very, very critical to have an overall approach to neutralize all these toxins. So rabies is another serious problem which requires attention. Basically, almost every 30 minutes, there is a death occurring due to rabies. 40% of the bites are in young children, and less than only 7% of these categories, in fact, of category 3 bites get treated with RIG. And it's a major issue, and WHO intends to eliminate rabies deaths by 2030. Just to give you a brief on the manufacturing processes which govern this. So you start with animals, and the animals are screened for various viral markers or bacterial diseases as per OIE, as per the WHO. And once they are actively checked by either PCR or by other ELISA tests, then they are quarantined for a period of almost around 3 weeks to 4 weeks. Subsequently, they undergo immunization process. So this immunization could be various types of immunogens. It could be venoms of Indian origin. It could be rabies vaccines for producing rabies immunoglobulin, or it could be thymocytes for producing the immunosuppressant. Then the antibody is estimated in individual animals. Subsequently, plasmapheresis is done. The animals are bled. The red blood cells are given back again to the animal so that the hemocretinol is maintained. And then the product is diluted with physiological saline. Preservatives are added. Enzymatic digestion is carried out to ensure that IgG is broken down into the FAP2 portion, which is the antigen binding arm, and the FC portion is completely removed. And subsequently, precipitation is carried out using caprylic acid, or also known as the octanoic acid, in which the immunoglobulin fraction, which is the critical part, which is the FAP2 region, remains in the solution, and the rest of all the proteins go into the precipitate. This is separated by centrifugation. Ultrafiltration is carried out, column chromatography, followed by nanofiltration to remove any viruses which may be coming of any animal origin, then followed by ultrafiltration again and the QC tests, and finally freeze drying and capping. This is a typical process for animal-based immunoglobulins. For human-based immunoglobulins, also the same similar process is employed. Instead of caprylic acid, some of the manufacturers may use polyethylene glycol or alcohol for precipitation, and enzymatic digestion is not carried out for the process. There are many steps which are undertaken for during the process in which the manufacturing is carried out. So during immunization, the fitness certification of the animal should be critical. Observation period, there should be 3 days. Immunization records, you need to have a complete profile of the animals, LFT, KFT, hemat profile, the titer testing to see what is the titer in these animals, weight of the animals is recorded, and subsequently sick animals should be removed from production the moment you have any of these parameters going out of the process. During plasmapheresis, again, similar profiles to be maintained, animals to be observed at least for 24 hours to see whether they have post-plasmapheresis reactions, because many a times when you do transfusion, you may have transfusion-related reactions in these animals. So that's very important to observe them again. So one approach is to look at the regulatory framework, which is, I would always say, the devil lies in the guidelines. So unfortunately, our regulatory guidelines are a little obsolete. And if you look at international guidelines, you see the international harvesting volumes for humans as per extrapolated to horse body weight of 225 kgs. They allow anywhere between 104 to 257 liters if you extrapolate the body weight. If you look at publications on horses, it is between 103 to 132. You look at our guidelines, which were made in 2001, it is 20 liters, which is far lesser than what even humans can give. Now, the impact we have to understand when such guidelines are made and they are not changed with time. If you look at it, the companies, they would prefer 54 liters, which would yield them approximately 1,900 vials. 53 equines would be used. But when you make such guidelines, you are utilizing the same animal and you're using 26 liters from that. Now to get those 1 lakh vials, you are actually using 110 animals. So basically, you are utilizing twice the number of animals because the guidelines are wrong. So it's very important to amend the existing CPCSA guidelines. This also means it puts a reverse effect. The immunogen is basically venoms. So venoms come from the snakes. So again, the snakes will be put in captivity and they will be milked for these venoms. So you are utilizing double the number of snakes for immunization. So without compromising the health of these animals, you can actually minimize the number of equines and maintain the same demand and ensure that all the patients are serviced and they are treated. In rabies anti-serum, there are various processes. So one way of looking at it would be improving the reach, and which meant a large scale production processes, which would mean lesser number of batches coming for testing. So bigger batches, smaller testing requirement. Also, effective use of this increasing the yield. So when you increase the yield, what happens is the total animal footprint comes down drastically. So you detect the low responders. Many animals, they don't respond effectively. Almost 20% of the animals don't respond. Detect them early, remove them from production, let them be rehabilitated, use only effective animals. No point in blending or bleeding those animals. That's cruelty. So high specific activity antibodies, you have to focus on, many of the manufacturers have to focus on the way the immunization processes are carried out. So you need a very high specific antibody, which also means the high activity antibody, high the titer, lower the protein, which means lesser the number of adverse reactions in the patient. So process development plays a key role, and you can work on the downstream processes to increase the yield, upstream processes by immunization, and keeping the annual production constant, but the numbers lower. But the permanent solution for this is actually the recombinant approach for rabies. And some companies have successfully tried and tested it. It's expensive, it's time consuming, but it's definitely possible. Coming to the snake venom antiserums. So here it's very critical. Each venom component, as I showed you in the beginning, has multiple venom fractions. And every animal doesn't show a similar reaction to all these venom components. As a result, what you have to do is you have to follow a flexi protocol. So you have to immunize the animals and test the titers in individual animals to see the responders, non-responders. And the non-responding animals, again, have to be removed from production. If the venoms are excess and the antibody titers are reached and the antibody titers stable down, you have to stop immunizing those animals. Immunize only animals till they reach a plateau. So one protocol doesn't fit for all the animals in snake venom. This ensures that whatever morbidity is generated by giving these animals this immunogen, that morbidity is reduced drastically. So this is something where the regulatory also comes in. All this needs to be documented. So many of the manufacturers prefer to do one single protocol for all the animals, which needs to be changed. So rehabilitation of non-responding animals, as I mentioned earlier, process development to increase the yields. This would help in decreasing the venoms and the snakes by almost around 50% and decrease the number of equines required for the production. What are the alternate methods? The recombinant approach. However, there have been difficulties in the recombinant technologies because you have multiple epitopes, multiple targets against which antibodies have to be made. Some of the positive research articles which have come up, although they have not been clinically tried and tested. So you have the androctonus scorpion in the Middle East, which has been tested for the antibody generation by monoclonal antibodies. You have the Crotalus species of snake. You have the centroidus antibody. Then the honeybees they have made. The mambas, Dendroaspis polylepis, there against the dendrotoxins, there is a monoclonal antibodies which have been made. Similarly, against the alpha and beta toxins from the Liaris scorpions. So several small molecule fractions have also been tried and tested. They have given some results. And plant extracts, if you see many of the toxinology society papers in India are showing that plant extracts are also effective against snake venoms. But the question is, when it goes to clinical trials, nothing reaches after the preclinical studies. So some of the smaller molecules wherein clinical trials are ongoing now are varispladid and prenomastat like molecules, wherein these have already been tried and tested for sepsis and for cancer. And they have shown very active inhibition of certain venom proteins. But I strongly believe it is going to be a combinational effect wherein you may have to use some small molecule fractions, you may have to use varispladid, prenomastat, you may have to use some monoclonal antibodies and then finally make a cocktail to reach such a big repertoire of antigens to be neutralized. So when you make or develop antibodies in animals or you use them for research, very important to ensure that the feed composition and intake, which is the dietary fiber, the nutrients, all that has to be very, very critically looked at. The type of immunogens for immunization, you have to look at it whether venoms or viral vaccines, human cells, you need not give active bacteria into these animals when you are immunizing them. The adjuvant schedule and immunization dose is very critical. You cannot give a very high dose, otherwise the toxicity will kill the animals. All the venoms are toxic. There will be liver toxicity, there will be kidney toxicity, so you have to be very careful with the doses which are given. Type of adjuvant use also makes a huge difference, because many of the prunes complete adjuvants and all use Mycobacterium tuberculosis, which can cause huge abscesses and morbidity will be very, very high. And bentonite, aluminum salts, the size of the dosage, the amount of dosage, very, very small doses in small proportions, multi-site immunizations with effective 3 to 4 weeks rest between each immunization, all this is critical. Bleeding volume so that the body starts regenerating for the next set of cycle. Frequency between the two bleeds, rest period between the two cycles, antibody titer is very critical, because if you have excess antibody circulating and if you still continue giving the antigen again, what happens is the immune complexes are generated and there's a feedback inhibition which suppresses the immune response. So all these are critical. And key health parameters would be, as said earlier, hemoglobin, LFT, KFT. There has to be a cutoff for the animals before bleeding. The hemoglobin content, the hematocrit has to be checked in the animals before bleeding them. And finally, we come to rehabilitation. So rehabilitation is, it also means that we are responsible towards these animals. And why do the animals need to be rehabilitated? So first is project completion or cancellation. Once you finish the project, they have to be humanely rehabilitated. They may have a poor response. They may be non-responders. They may have chronic debility. Bilateral opacity is seen many a times when you give venoms and other toxins to these animals. Arthritis is seen because immune complexes start depositing in the knees and other joints of these animals. You may have poor hoof condition in some of the animals, like canker and other diseases which may hit them. Old animals, after a certain time, what happens is these animals, the immune system starts going down. So that's the time when you need to look at rehabilitating these animals. But when you rehabilitate, your responsibility doesn't end there. So you need to be knowing that you have to check the NGO which is going to do this. You have to check whether they have enough land, animal shelter. They should see that the animals are free from sun, rain, winter. They are rehabilitated properly. Water and feed is there, veterinary care, dedicated staff, and it should have funds for running. Many a times you may send it to an NGO and that NGO may sell that animal back again to somebody else. So you need to see end to end for these animals. And then it is quality attributes. So the focus has always been on replacement, reduction, and refinement. So you can go for replacement, could be cell culture techniques, isolated cells and tissues, mathematical modeling. In reduction, you minimize the number of animals based on public data available. In refinement, I would always say you can reduce the pain and suffering when you're doing the testing and all. But secondly, why not go to somebody who already has the necessary infrastructure, who has the set of guidelines, who has IEAC permissions, who has CPCSA approvals, who knows how to do these experiments? So use public testing laboratories who have these facilities, rather than opening your own facility and doing the testing on a regular basis. Coming to the quality control assays. So one would be abnormal toxicity assay, wherein the Indian Pharmacopoeia Commission, and we are working very closely, the manufacturers, and PETA is working very closely to ensure that these are removed from all the monographs. Slowly, one by one, most of the monographs are deleting these abnormal toxicity assays. And unfortunately, the currently available models have limited predictive value, So especially giving an example of, if you're doing an anti-infective antibody in an animal, you have to know that the baseline of those antibodies has to be absolutely zero. But sometime or the other, these animals must have undergone infection and those antibodies will already be there. So selection of the animals right in the beginning is very, very critical. One next thing is on the potency test, where we've been working very closely, manufacturers, PETA, as well as the IPC, the government has been working very closely. To give an example is the rabies neutralization test. So this was developed by the NIH in 1960s. Subsequently, in 1984 came the plaque reduction assay, which is the first in vitro assay. This was superseded later on by the fluorescent focus inhibition tests. When finally the RFFIT is now the method of choice with very good sensitivity. And almost around 10 years back, it has been in all the other pharmacopoeias. Indian Pharmacopoeia, we all the PETA, the manufacturers, as well as IPC is working closely with CDSCO to change this particular guideline. So this would save a lot of animals when we go for cell-based assays. Similarly, for other methods, so you can, for tissue culture models, cellular systems, you have your MAT as Ashwin mentioned. Then you have animal biomarkers wherein you can do QSAR, you can do molecular docking studies, microfluidic biochips are available. And most importantly, what you can do is you identify such lab tests. That's very important, which are not in line with yours, and then try to eliminate and replace them systematically. Then you have various methods which are already validated by Altex, ICCVAM. Use those methods in your regular safety testing. Some of the disadvantages are may not mimic the biological outcome in the whole body. It is also possible long-term experiments and outcomes cannot be studied. Use of cells, their interaction, differentiation depends on feedback of metabolic cascades. These are some challenges which the investigators have. And finally, I feel the ethical integrity is just not the ours, but it is your honesty with which you are going to do this work, your social quotient, your animal welfare quotient, and your scientific integrity. So it's a harm benefit analysis you have to do. And it's this entire moral compass which is there, which is very critical when you design your experiments. That's my team. We work together on multiple issues. We work with molecular biology, we work with nucleic acid purification, we work on QC platforms. That's a 25,000 square feet we have in Bangalore. We have 2 facilities in Bangalore, and at 2 different locations. We are ISO certified 13485, ISO 9001, we have CDSCO approvals, and we strongly believe in Make in India concept. At last, animal ethics is as important as human welfare. Let's not forget that. Thank you.
Thank you very much. And I understand that online, Dr. Kalivani is online, right? Wonderful. I think you can just request her to be there.
Good evening to all. Hope my slides are visible and I am audible. Yeah, good evening to all. Hope my slides are visible. Yeah, am I audible? Once again, good evening to all, and I thank organizers and PETA for providing this opportunity. And here, this is brief about what are all the activities related to alternatives to animal method at Indian Pharmacopoeia Commission we are focusing on. So this is our Indian Pharmacopoeia Commission. Yeah, Indian Pharmacopoeia Commission is an dedicated autonomous scientific institution under Ministry of Health and Family Welfare. The important mandate of Indian Pharmacopoeia Commission is to publish Indian Pharmacopoeia, which is a legal official book of standards for drugs and pharmaceuticals approved for marketing in the country as per 2nd schedule of Drugs and Cosmetic Act. And Indian Pharmacopoeia specifies standards of quality for identity, purity, and strength. And Indian Pharmacopoeia Commission was operational from the year 2009, and it works with the guidance with guidance from general body, governing body, and scientific body. And we have IPC Secretariat and Indian Pharmacopoeia Laboratory at IPC. When we talk about Indian Pharmacopoeia, as I said earlier, it is a book of standards, and it prescribes the standards for quality, identity, purity, and strength. And it is legal as per second schedule of Drugs and Cosmetic Act. The Indian Pharmacopoeia is accepted in 4 other countries. Other than India, it is accepted in 4 countries. And Indian Pharmacopoeia Commission is a member of Pharmacopoeia Discussion Group. The Pharmacopoeia Discussion Group is a group which has USP, United States Pharmacopoeia, European Pharmacopoeia, and Japanese Pharmacopoeia as a part. And now they have included Indian Pharmacopoeia as a part of this group. Yeah, this is the journey of Indian Pharmacopoeia. Like, these are all various editions of Indian Pharmacopoeia. The current edition is Indian IP 2022, which has 4 volumes, and it contains more than 3,000 monographs of all aspects like small molecules, pharmaceuticals and biological products, vaccines, blood products, phytopharmaceuticals, and veterinary products. And this is the monograph status. As I said, it has more than 3,000 monographs of different categories. And to make these monographs in Indian Pharmacopoeia, we have a network of more than 150 scientific experts from industry, from regulatory authority, and from academia who are involved in development of this monograph, other than IPC scientists. And when we talk about alternative to animal method, IPC adopts any of the following, adopts various approaches. The approaches are comparability and applicability suitable.
Sorry, Dr. Kalmani, the slides are not moving.
Okay. Can you confirm? Is it now?
Yeah, it's working now. Yeah, perfect.
So when we when we talk about alternatives to animal method in Indian Pharmacopoeia, like IPC adopts certain strategies. The first one is comparability and applicability of suitable non-animal method or test in place for current in vivo method. So the first, we look for the comparability and its applicability for this animal method, which was already in IP. And alternative approaches are also adopted based on various scientific literature and based on analysis of retrospective data, what we have done in abnormal toxicity test and target animal batch safety test in veterinary vaccines, and good manufacturing practice and pharmacovigilance in place. By considering all these aspects, alternative approaches are given that I may explain while I am explaining the abnormal toxicity test approach. And implementation of consistency approaches are considered, and international harmonization with regulatory requirements like WHO, technical report series, and other pharmacopoeias. Other than this, we receive expert opinion from various experts involved in this alternatives to animal method research and area, for which IPC have constituted an expert working group for alternative to animal method, which involves various scientists from CSIR, ICMR, and industry. This was already said by Dr. Rohit, that IPC have constituted this committee and all. Yeah. When we see non-animal methods in Indian Pharmacopoeia, there are, we can group it into these 4 categories. The first one is the animal method for determination of potency in most of the biological monographs like vaccines, biotechnological products, and blood products. And abnormal toxicity test, specific toxicity test, and of course pyrogen test, which is there in all parenterals, and the target animal batch safety test or laboratory animal batch safety test in veterinary vaccines. So our focus is on all these 4 areas. When we see about abnormal toxicity test, as it was already said by Dr. Alden and Dr. Rohit, that IPC have removed abnormal toxicity test, or yes, in IP, a note has been introduced stating that the abnormal toxicity test may be omitted for routine lot release once consistency in production has been well established up to the satisfaction of National Regulatory Authority. That means each lot need not to be tested for abnormal toxicity test. And of course, this abnormal toxicity test is stated in the specific monographs of vaccines, but it may be omitted if the consistency is established up to the satisfaction of National Regulatory Authority. And we are working with the stakeholders to understand for complete removal of abnormal toxicity test from the specific monographs. And similar type of approach was considered for specific toxicity test in diphtheria-based vaccines, which impacts almost 16 vaccines, where the specific toxicity test in the final bulk can be omitted for routine lot release once consistency in production has been established. And this is what with respect to toxicity tests. And when we see about test for pyrogen, and it was already said by most of the speakers that pyrogen test has been removed or replaced with the bacterial endotoxin test in most of the parenterals. And few biologicals like blood products and certain vaccines where the complete removal is not possible, an alternative approach is given. Showing here like pyrogen test or bacterial endotoxin test if justified and authorized by NRA. Based on validation of the test. If any manufacturer wanted to show their validation and prove that bacterial endotoxin test is better than pyrogen, then it is, as per Indian Pharmacopoeia, it is acceptable for it. And such type of approaches are also given wherever the complete removal is not possible or replacement is not possible. And a monocyte activation test general chapter has been introduced to promote the stakeholders. If interested stakeholders or interested manufacturer can adopt this monocyte activation test too, but it is not included in the specific monographs of Indian Pharmacopoeia. And during discussion, there are various challenges with respect to monocyte activation test, like it has different technology and which particular technology needs to be adopted for which particular product. That clarity is lacking among the manufacturers, and the diagnostic kit cost which is available is very high, and it needs hands-on training, like stakeholder needs hands-on training on these technologies. And there are no or very less laboratories performing this test. And apart from this monocyte activation test, and recently we have received the proposal for inclusion of recombinant factor C or recombinant LAL as a replacement for bacterial endotoxin test, which is under discussion with the industry stakeholders to understand better what are all the challenges and before putting any monograph or general chapter in Indian Pharmacopoeia. And next, when we see that the major one is determination of potency using in vivo method, where the replacement, refinement, and reduction has been considered. Certain in vivo methods, wherever possible, if there is comparable in vitro methods are available, and those in vitro methods has been in vivo methods are replaced with in vitro methods. And yeah, and in biotechnology derived products and all, in vivo methods has been replaced with in vitro method or by analytical based methods. And wherever complete removal of in vivo method is not possible, an approach has been given that in vivo animal based method during development stage, once consistency established, in vitro potency can be adopted. So such type of provisions are also given so that manufacturers can adopt the in vitro based method too. And in the aspect of refinement, certain in vivo methods are replaced with serological based method, where serological based methods, and certain in vivo methods wherever the multiple dilution methods are replaced with single dilution methods in few of the human vaccines and immunosyrup for human use. And the next one is target animal safety test, batch test safety test, where a provision has been given based on the retrospective analysis of the data, which are available from the domestic and international manufacturer, and based on VICH guideline. And this particular approach has been introduced very recently in Indian pharmacopoeia, that batch safety test using target animal may be omitted if safety test has been performed with the satisfactory result in master seed lot. Consistency of manufacturing process has been well established up to the satisfaction of NRA, and at least 10 consecutive production batches has been produced and comply with safety test. Significant changes to the manufacturing process may require resumption of routine safety testing to re-establish consistency. So with this, every lot which was tested for target animal batch safety need not to be done. It can be, manufacturer can omit such test in the every lot. And apart from these things, IPC introduced a general chapter on substitution of in vivo method by in vitro method, a guideline. It is a general chapter which gives the guideline for substitution of in vivo method by in vitro method for quality control of vaccines. And there is a guideline for the use of alternate test to bacterial endotoxin test. So these are all the general chapter or guideline which are introduced in Indian pharmacopoeia. Yeah. These are all the things which IPC has considered in the area of alternatives to animal method. Once again, thank you for the opportunity.
Thank you very much, Kalayani and Dr. White for speaking here.
Yes, thank you, sir. Good evening.
How are
sir.
you?
Fine, sir. How are you?
Fine. Nice
Good,
to
yes,
see
sir.
your
Good to see you
presentation.
here. Yes, sir. Thank you, sir.
Wonderful. So I think we
Thank you,
had
sir.
the four wonderful presentations, starting with Dr. Rohit, and then Dr. Eldon, and Dr. Bhavekar and Kalaivani. All four, I was never knowing that they will be such a wonderful presentation, but I pity that those who are not able to listen to this is a loss for them. And all of them are really directly working towards our objective, what we feel. So maybe I will just raise some question. Maybe the first one with Rohit. Rohit, you mentioned about the replacement of animal. I think what you have mentioned is in the broader sense. So, and to all of you, all three of you, and also to Kalaivani, see, all of you have suggested that there are several methods which in regulatory system have already been in the process of being introduced. Like is it going? Like Kalaivani has also mentioned that these are the guidelines which have been mentioned in Indian pharmacopoeia, in the pharmacopoeia, that these are the guidelines. So my just question is how we can fast track this process. A, identifying that, yes, say for example, you mentioned about the snake venom. This was a task force in DBT, and I was part of that, and to foster, to speed up the process of snake venom. There was a task force in ICMR for rabies, and that how we can, and instead of having, why not we have this? So where is the bottleneck? Is this the industry does not approach them, or you think that the Pharmacopoeia Commission Scientific Committee should approach the manufacturers that these are the things? So where is the synergy where we can do that? Because we have, all of you have very rightly identified several areas, critical areas, I would say. So what do you think?
Hello. Yeah, I'll answer that.
Yeah, all of you can answer.
Yeah. So a few things. So one is with the respect to the production, as you mentioned. For each one, like say for rabies, there was already technology available with the WHO, and some companies took advantage of that, and they made their own monoclonals. It's not fully humanized, but it's a mouse-human chimera which is being used. So there are alternatives. But the question is, what is the time? What is the cost? Can the Indian patients afford that? So there is room for, in the developing countries, there is room for both. There is room for the low-end equine products, which are good in quality. Also, there is room for the recombinant products because only 7% of the market need is being met right now. So saying, the time required to bring a monoclonal into the market is almost around, as you know, 7 to 10 years is minimum which we are looking at. Now, when you go to develop, this should have been done long ahead, before time. But if these products are under DPCO and the cost of these products comes to 200 rupees or 300 rupees per vial, which is not affordable to the common man. So the common man still wants something which is cheaper, which goes straight into the equine category. Whereas somebody who's affluent, then they take the monoclonals and other products which they can utilize. So these company manufacturers are not willing to shift from the equine products to recombinant because they neither have the funds which are coming in. So if DBT can help these manufacturers directly and work together, I'm sure the manufacturers will be willing to take this, but it will be another 7-year horizon wherein the products come in. That is for rabies, say for instance. But so saying, there are 2 big pharma companies which are already having their recombinant products in the market. Second thing is, and slowly there's been a paradigm shift. So now it's equally balanced between equine and human. There are 2 products already, rabies in the market. Now the human also is imported by, say, Grifols or CSL or somebody else, because the Indian guidelines do not allow the candidates in India to be immunized and their plasma volume to be extracted for any of these, be it for rabies, be it for hepatitis, be it for anti-D. So the guidelines do not permit. So we are 80% dependent on plasma coming in from the US for human blood products. That's the sad part. And second thing is with respect to the equine. Now equine and human are balanced. And the third is the recombinant. Slowly recombinant is increasing as per the affluence of the people. And slowly there will be a shift. So these products will ultimately meet a death scale in another 5 years or 6 years or somewhere down the line. So it is always in the interest of the government as well as this thing to have multiple manufacturers, support these manufacturers to transition from animal-based products to recombinant products.
I will just, because there are a lot of points, but you have made very important points. Serum, equine, human, or recombinant. There is a logarithmic increase, if not logarithmic, but there is a tremendous increase in the cost of recombinant as compared to conventional bovine. And this was also discussed a couple of forums that there is efficacy wise, there is not that remarkable difference between the 3. However, there may be some incremental benefit in terms of allergenicity and other side effects in terms of recombinant. But if you look for the tangible benefits in terms of reduce the number of equine or maybe the animals which are used is a indirect evidence. So this will be de-risking, or maybe the NPPA or DPCO, how long they can support this. I think the price of the recombinant has to come down ultimately. Otherwise, this would be, I don't know, this will remain the. I have a question with Rohit. You said that there are several already standardized products available for Epi Ocular, Epi Skin, Epi Derma, Epi couple of things. But all they are trademarked, all they are commercial.
Yeah, they are commercially available.
So you have to buy them.
Yeah.
So there's a lock and key system. But that means they can earn money, and then this is a company centric. OECD has approved it.
Yeah.
There is no generic version. What is this dilemma? Why should I take it? Because this is already company, is making money, and OECD has approved it. I have to use it, I have to buy it. So can there be something which Indian startup maybe with the funding of DBT or BIREC can make the same preparation, make in India? This may not be rocket science, I do not know. So हम क्यों नहीं बनाते हैं इसको अपने आप?
So actually,
हम
the
FET पे depend करते हैं.
funds. Second thing is the hand-on experience. Sorry? Hand-on experience. Like
people like experts like him, experts like you.
Yeah, means like, for example, like biodimension technologies. These guys, they are now started developing these three-dimensional tissue models. They got funded from DBT, BIRAC, CSI, and other funding agency. So they are now getting funding from BIRAC. Nowadays, means if you also know, sir, now BIRAC has also the funds are rising, they are also promoting the startups more and more. And now there is this recent amendments in the new drug and clinical trial by Ministry of Health and Family Welfare. They have included this all preclinical testing, these alternative models in preclinical testing of new drugs and investigational drugs. Now they have agreed and accepted these alternative test methods should be included in preclinical testing. So these type of initiatives also encourages academic scientists and industrial scientists. Now these are under the regulatory guidelines. Now we can move it. So now the academic, it means this is the one example of a startup. But if we go for IITs also, like ICT Mumbai, they are working like phenomenal in organoids. And some of the organic model, these models are under validations, and soon they will be go for the regulatory approval or the communication with the regulatory bodies for their approval. So these things are now they are coming up. They are accepting and they are understanding the value of alternatives. And obviously, some of the models, not some of the majority of the developed models are of Western countries, and they are very, very costly. But now, like monocyte activation test, some of the Indian companies are now developing the in-house monocyte activation test as an alternative, and they are trying to validate them. So now the companies are also, but these initiatives are very limited. Companies are taking these initiatives, But once it is established by the one company, I think that is an example for other companies to follow, and that's how these things will be changed. For example, like recombinant technology, I think the Serum Institute of India is a pioneer in it in rabies. They have already have the recombinant monoclonal antibodies for rabies. Now we are communicating with other biopharmaceutical companies to collaborate and submit a funding proposal to DBT and BIRAC to develop some monocyte. And we have already discussed with DBT BIRAC on this, and they agreed that you can submit and we will see. Like we are also promoting monoclonal antibodies. Like recently, there's a news and it is also projected in the social medias that BIRAC in his national biopharma missions, they are also doing meetings and promoting and doing meetings with the stakeholder companies to promote the monoclonal antibodies to move away from this convention. So things are going on.
Great. Dr. Kalaimani, just one thing. See, I will just a nostalgic feeling which I had when I joined AIIMS as a lecturer long back in '83. My head of department gave me one job that you are tomorrow, you immediately that you will be in charge of pyrogen testing. And I used to inject into the ear vein of rabbits and then put a cannula thermometer and do the pyrogen testing. And all the samples which used to come from the hospital were released after. I used to feel that I am running the hospital because after my signature, the sample will release. Now, those were the days when the pyrogen testing was done as per pharmacopoeia. So then I realized that there is something government called as a pharmacopoeia. Before that, I was not doing the pharmacopoeia. Kalaimani. Now, just tell me, Kalaimani, how and who will approach you? Who, in this case, who approached pharmacopoeia that this is to be removed? Is this the PETA, or is the scientist, or is the initiative of IPC itself? And that is a case study. Same maybe for 4 or 5 which you have mentioned. So who will take initiative? Is the PETA is one body? PETA may take 1 or 2. But can this be? So you can identify many things. So where the ball should start rolling? Over to
Sir, yes, sir. Thank you, sir. Sir, it is all has to do, but it based on the test which, like for example, in case of monocyte activation test, it was of course a PETA's initiation, and because of PETA's proposal, we have considered that, and that was discussed with the manufacturers, and then we have introduced the general chapter. Whereas in case of certain alternate methods which we had given in the biological monographs, it was the manufacturer who had proposed the amendment. It was the manufacturer who have proposed the amendment along with sufficient validation data and all. And that has been considered for discussion and verification at in-house laboratory facility, and that has been considered. Whereas certain analytical methods, which, for example, in case of in case of insulin and all, like NIB had proposed as this HPLC based method is better than what was existing. This was earlier days, I'm telling, sir. So now that HPLC based method is there in Indian pharmacopoeia. So this is all the ways how, like, if we receive a proposal, if IPC receives a proposal from any of the stakeholder, it might be an institution, it might be an industry, or organization like PETA and others, like we take up and we study, and then the decision was taken.
Incidentally, I am fortunate to know Kalaivani for last almost 20 years.
Yes, sir.
Having an officer of her caliber in IPC is I think a wonderful thing. So just
Thank you so much.
just tell me one thing. You told me that in this case, PETA took the initiative, right? And then,
Yes.
of course, followed by. How did you reward PETA?
Sir,
No,
it
see,
has.
there is something which, see, I am very frank with PETA. The PETA was not considered as a good word in scientific community in majority of the institution. And जब PETA का नाम लो ना, तो वो बोलते हैं कि सिर्फ फ्लैग लेके सड़क पे जुलूस निकालते हैं जहाँ एनिमल का एक्सपेरिमेंट होता है. But here you see the different aspect of the coin, which you should make popular. I think you are doing such good for science and society, and also for the animal world. But perhaps अभी आपका ना वो tag उसको हटाना चाहिए, and then that's why I'm saying, how did you reward PETA? So that you should have popularized कि PETA ये भी करती है.
Yes, sir. अच्छा, शुरू में हमने भी ऐसे ही सोचा, like PETA came, why they are coming, all these things. And later, based on their proposal, when we could do something good for the animal welfare, then we realized it is good work, the proposal which PETA is giving. So, of course, sir, reward in terms of, we consider them in every expert meetings, discussions, and wherever I go with respect to alternatives, I used to refer them. So such type of things which I can do. This is what.
I will tell you my experience with the PETA was very good. And in fact, at that time, my director was Dr. Dekha, and then there was a big news item कि एम्स में बंदर केज में से निकल गए. Oh, you remember? You may not be there at that time. And then it was Menka Gandhi who summoned us. The director has no courage to face. So he said, गुप्ता को पकड़ो, तुम जाके झेल के आओ. This is word, तुम जाके झेलो उन्हें. So I went, and she became my good friend even today. That is a different story, but that's how. So just tell me one thing. ये पायरोजन टेस्ट तो आपने हटा दिया. अब दुनिया भर में कहाँ से आता है मोनोसाइट एक्टिवेशन टेस्ट? Is this done by everybody, or still people, because pyrogen test is not banned. It has been removed from pharmacopoeia. But many labs, I believe, are still doing it. So is it true, or it is not true? Maybe Kalaivari can tell us.
Sir, certain products today also it needs pyrogen test. So those things needs to be performed. Even in pharmacopoeia also, certain biological products, we ask for, like it says, for pyrogen test only. Maybe those products, those laboratories might be doing.
What is your...
Yeah, I think what Dr. Kalaivani said, it's right. So you're coming back to your second question, like European pharmacopoeia, they come up with the 2010, so 13 years back, and popularly this test is done in the European continent. Japan has done extensive validation. And coming back to the first your question regarding the bottleneck, regulators are really encouraging us to implement with the manufacturers, but we are really waiting for the local data. As an Indian government, I need to submit the data. I have the validation data right from my principal company that I cannot straight away use. Even I have the extensive validation in Japan, but that I cannot until and unless I will establish the data here. And that data is expensive to establish. As a manufacturer, we cannot sponsor certain number of kits due to the cost issue. It's not like media sample, we will sponsor, say for example, 10,000 test samples or 13,000 test sample to establish that data. So biggest bottleneck is the cost. And second is post-COVID, there is a stringent regulation on certain custom aspects. So when we ship the cells from France, Holcim site, so it reaches to India within 2 days, but another 2 days it will take customs. So in that case, certain product aspects, especially cells, other products are stable, like endotoxin pyrogens and non-endotoxin pyrogen that are the stable entity that require 2 to 8 degrees centigrade. But the biggest problem is the experiment is the cells that are stored in the DMSO, and by that time it released from the custom and reaches to the customer site, cells get died. So that is one of the biggest technical bottleneck I wanted to highlight.
Cost, custom, and the feasibility. So if I recommend Kalaivani, then why not? Because you have now a chapter. This is my last, because you have got an indication that you have to wind up. Otherwise, I have so many interesting questions. Maybe we can, if they have planned for dinner, we can have that till midnight. But my one suggestion to Kalaivani, because there are many things which Indian pharmacology is one. Because you have a chapter. You said that you have a chapter of guidelines for incorporating the in vitro or alternate in this. But there are several things which are, I would say, low hanging. Where one he's saying that it is because of NPPA, DPCO, and he's saying because the custom, because it's, and then the same thing is being like you have an everything epi, but there is no Indian product which can make it cheaper and available. So why don't you organize a brainstorming by IPC involving all these people? I can talk to Dr. Raghuvanshi and then have, which is something which the government can be sensitized. And yeah, ये कर रहे cost पे कितना हमको फर्क पड़ रहा है? How many? Because there are a lot of production linked incentive and the research linked incentive. Why not these things? And so maybe we can sensitize. So, and you can use this forum, and maybe I will take final advice from Dr. Raghuvanshi and Dr. Aishuddin. We can use this forum and we can jointly organize this. What do you think?
Yes, sir. Your advice is taken, and we'll discuss with our director.
Maybe these are the treasure of ideas. We can just give not many, 10 ideas. Then these are the ideas where we can look for. Because IPC is very, very, very fertile and very active body.
Yes, sir.
And they face a lot of court cases, lot of representation, lot of inquiries. They're used to it. Because I know that race test was there. That was also started by this, and then there is a diethylic. What is that? The excipient in the diclofenac injection, where it was introduced into the pharmacopoeia. It was taken out, then it was introduced or taken out, and it went to court. So they are used to it. So take home message is take this initiative.
Sir, when an organization works, then we'll get questions, and these legal issues will be.
That is what the science is.
Yes, sir.
You are known for it, scientific rigor.
Yes,
so
sir.
Your name, at least we will write in golden letters in history of this society. So I think we should do it.
Yes, sir. Sir, before that, we need to have a discussion with our director, like Dr. Rajiv Singh Raghuvanshi. We'll have a discussion, initial discussion, and then we'll take up this.
But you agree to take this to director?
Yes, sir. I'll take definitely. After this conference, I'll discuss. When you say, then I cannot miss that news, sir.
Just we can have maybe.
Yes, sir. I'll take up.
We should have this. This will
yes.
be very. Thank you very much. Sorry, my apologies. Any question and comment from the floor? Last 1 minute, because she has already come to take mic from me.
Thank you, sir. Thank you all the speakers for the wonderful session. I'll now request Mr. Dr. Y.K. Gupta to hand over the token of appreciation to all the speakers. Dr. Rohit Bish.
All four did wonderful presentation.
Dr. Ashwin Kumar. And Dr. Alden Fernandez. I'll now request Mr. Dr. Ashwin Kumar to hand over the token of appreciation to Dr. Y.K. Gupta, sir. All right.
Kalaimani, are you hearing?
Yes,
I'm
sir.
taking your memento.
Sir,
And if I
your
remember, I will
appreciation
hand
is
it over to
important
you.
for us, not this one, sir. I'm happy that I'm with you in this panel discussion. Thank you so much, sir.
Thank you, sir.
Thank you, Kalaimani. Thank you.
Thank you, sir. Thank you so much. Thank you so much. And thank you all organizers too.
Thank you, sir, and thank you, ma'am, for this wonderful session. Next will be an informative visit to Ibn Sina Academy, for which bus has been arranged outside. Okay, sir. Which will be followed by we will gather here for the general body meeting. It will be followed by cultural program. Thank you.